마이크 테스트 하겠습니다. 마이크 테스트 하겠습니다.
잠시만요.
네. 좀 소리 들어오는 게 정상이에요?
지금 BGM이 들어오고 있긴 한데 엄청 조그맣게 들어오고 있어서
네.
안 들리실 수도 있어요.
아하. 운동연구 센터 구축 사업 총괄 심포지엄 2025를 곧 시작하도록 하겠습니다. 장내로 들어오셔서 착석 부탁드립니다. 오늘 행사는 지금 2일차인데요.
Opening remarks followed by the session and it will conclude the conference. And today's symposium will be conducted in English with simultaneous interpretation service provided. So please feel free to use the receiver at the back of the conference hall or on the desk. Channel 1 for Korean and channel 2 for English. And in the Square Ballroom lobby, you can enjoy various programs including Regional Achievement Exhibition, a Vision Promotion video, and a photo zone. We kindly encourage all participants to take interest and join in. And for lastly, to ensure the smooth progress of the event, please check once again if you set your mobile phone to a silent mode. And once again, please note that today's presentation materials are provided within an e-book format via QR code. You can scan the QR code on the monitor to download today's presentation materials. 네, 두 가지 안내사항 더 말씀드리겠습니다. 어제 등록을 하셔서 명찰을 발급.
And additional information, we have doled out the name tags and you are supposed to issue the name tags once again. For the second day event, please proceed to the registration desk and reissue your name tag. And also, you will find the QR codes for satisfaction survey. You can also find the codes on the screen, and we would like to prepare for an even better program for the next year, and that's why we're doing this satisfaction survey. And also we're giving coffee coupons for the participants, so we would like to ask for your active participation.
Ladies and gentlemen, may I have your attention, please? The Day2 program of the Advanced Bioglobal Joint Research Center of Symposium 2025 will begin shortly. We cordially ask all participants to enter the hall and visit it. This symposium has been holding for 2 days until today, and today's program will begin with opening remarks followed by sessions. Today's symposium will be conducted in English with simultaneous interpretation service provided, so please feel free to use the receiver at the back of the conference hall or on the desk. Channel 1 for Korean and Channel 2 for English. And in the Square Ballroom lobby, you can enjoy various programs, including regional achievement exhibitions, a vision promotion video, and a photo zone. We kindly encourage all participants to take interest and join in. Lastly, to ensure the smooth progress of the event, please turn off your mobile phones or set them into a silent mode. And please note that today's presentation materials are provided with ebook format via QR code. You can scan the QR code on the monitor to download today's presentation materials. Thank you very much and see you soon. Good morning, ladies and gentlemen, distinguished guests. Welcome to the Day 2 of Advanced Bioglobal Joint Research Center Hub Symposium 2025. And my name is Dae Jung, and I once again have the honor of serving as an MC for today's conference. And starting yesterday, Advanced Bioglobal JRC Hub Symposium 2025 has been taking place for 2 consecutive days. And it is jointly organized by the Ministry of Science and ICT, the National Research Foundation of Korea, and K-MediHub. And the symposium is held under the theme of Networking and Expansion of Global Joint Research Centers. And before we begin today's program, let me briefly announce the schedule. Following the opening remarks, we'll proceed with 2 sessions with 7 presentations. Once again, I'd like to express our sincere gratitude to all of you for joining us today. And with your big round of applause, let us begin the Day 2 of Advanced Bioglobal JRC Hub Symposium 2025. Now, to officially open the symposium, I'd like to invite one of our distinguished guests to the stage for the opening remarks. We were scheduled to have opening remarks from Dr. Gil-seng Jung, Director of the New Drug Division at the National Strategic Research Bureau of the National Research Foundation of Korea. However, due to unavoidable circumstances, he is unable to join us today. Instead, Mr. Hee-young Jang, Program Officer at the National Research Foundation of Korea, will kindly deliver the opening remarks on his behalf. And let's greet him with a big round of applause. I'd like to invite Program Officer Jang to the stage.
아무래도 둘째 날이다 보니까
This is the second day, and I believe that that's the reason why that the turnout has somewhat declined. I would like to briefly say hello to everyone. Good morning. I am Jang Hee-ung, Director of National Research Foundation of Korea. Mr. Dr. Gil-seng Jung was supposed to participate to deliver the welcoming remark, and due to other commitments he couldn't. We would like to ask for your understanding. This project started in 2024 based on the support from the Ministry of ICT in order to spur growth of biotech industry at home and abroad. And this is also to promote global collaboration in that field.
의 권역
At the moment, we have 14 regional centers and 1 control tower in place. And today, the research results and future growth directions will be shared, and that will be the major goal of today's event. Yesterday, we have listened to the speakers from Korea, Oceania, and etc. to spur exchanges about future growth. Today, we will listen to the speakers from European Union and to North America, and it will surely be an opportunity to exchange valuable insights. I hope that today's event will offer us an opportunity to spur growth of the biotech industry, and also I would like to appreciate the support from all organizers that have successfully organized today's event. Thank you very much.
Thanks once again. Mr. Jang extended a warm welcome and gratitude to all participants for joining us today. Please give him a big round of applause once again. Now, as we did yesterday, let's take a moment to take a group photo session with some of our distinguished guests to commemorate today's session. And may I invite some of our distinguished guests to the stage for a group photo? Yes, so since it is day 2 of the conference, we'd like to invite some of our distinguished guests to the stage for a group photo session, just like yesterday. And maybe we can do like a couple of poses. Can you move a little bit forward at the center of the stage, please? Look at the center of the camera with a big bright smile. It looks like we need one more distinguished guest for a group photo session. One more time with the fighting with your right hand. And can our participants give a big round of applause? 네, 여러분 혹시 박수 한번 부탁드려도 될까요? 오늘 두 번째 날 시작하는 날인 만큼
This is the second day of the event. We would like to ask for your big round of applause.
Now, this concludes the short opening ceremony of Day 2 of Advanced Bio Global Joint Research Center Hub Symposium 2025. And following the ceremony, we will continue with the main sessions and a networking coffee break here in the main hall. So we encourage everyone's active participation. And for a brief announcement for today's lunch, for participants, they can enjoy using the lunch coupons provided at the registration desk just like yesterday. And dining is available at restaurants located on this floor on B2 of the COEX Mago Convention Center. We look forward to seeing you again after you have the lunch session as well. And now, let's begin the first session of Day 2 of Advanced BioGlobal Joint Research Center Hub Symposium 2025. The first session will feature two themes from the North America region, biotechnology-based API production and the biofoundry. And each theme will include presentations followed by a short Q&A session. Before we move on to the main session, can we have one more group photo session just before? Because it looks like we have one more distinguished guest for a group photo session. Yes, before we move on to the main session, I'd like to invite our distinguished guest once again to the stage. Thank you very much. Because everyone knows picture is everything. 네, 여러분 남는 건 사실 사진밖에 없습니다. 오늘 둘째 날 기념하는
As you well know, picture tells all the stories. Picture is everything. So please look at the camera in the center and give us your best smile. And please give them a big round of applause.
with a big round of applause the Day 2. Thank you very much. And can we do the writing with the right hand once again? Yes, thank you very much, everybody. Thanks once again. And we can finally move on to the first session of Day 2 of the conference. And as I have explained before, today's session will feature two themes from the North America region, biotechnology-based API production and biofoundry. And each theme will include presentations followed by a short Q&A session. And for the first presentation under the theme biotechnology-based API production, and we'd like to invite our moderator who will lead today's session. We are pleased to have Dr. Mi Hee-jung from KIST. Please welcome her with a big round of applause.
Good morning, everyone. We are ready to start first section covering biotechnology-based API production. My name is Mi Hee-jung from Korea Institute of Science and Technology. My research focuses on development of gene and cell therapeutics through the gene modification to treat diverse diseases, including cancers and autoimmune disease. Especially, I'm working on developing mRNA CAR-NK cells by delivering mRNA through the electroporation, which is very simple and convenient ways providing potential benefits for large production of CAR-NK cells. To achieve this, I'm collaborating with professors from Canada. So moving on to today's presentation, it's my great pleasure to introduce Professor Seung-Han Lee from the University of Ottawa. He's a pioneer in NK cell-based research. During his PhD studies, he made a groundbreaking discovery by identifying the first NK cell receptor that recognized viral infection on virus-infected cells. Following his PhD, Professor Lee completed his postdoctoral training under Professor Christine Byron at Brown University, one of the most influential experts in NK cell biology. He began his independent research career at the University of Ottawa in 2011, where he now serves as a full professor from 2011 to 2016. From 2011 to 2016, he held the position of Canada Research Chair. Professor Lee's research has been published in top-tier journals, including Nature Genetics, the Journal of Experimental Medicine, and Cellular Molecular Immunology. His current work focuses on training NK cell biology into clinical applications, particularly through the development of CAR-NK cell therapies, the top of his presentation today. So please join me in a warm welcome to Professor Seung-Hwan to the podium.
Dr. Zhang, thanks so much for your kind introduction. So before starting my talk, I want to thank the organizer for giving this wonderful opportunity to introduce our collaborative the research, which is advancing mRNA CAR-NK immunotherapy through scalable manufacturing. So for this, the collaborative, the research, we are partnered with Dr. Zhang at the KISS, Korea Institute of Science and Technology. And then my name is Seung-Hwan Lee, and I'm the professor at University of Ottawa. So as you can see, this is the very peaceful, the summertime in Ottawa. Ottawa is, the capital of, the Canada. And then, the Canada is called, the country, that, right? So during the winter time, those, the all downtown, the Rideau Canal is transformed to the world's largest, the skate rink. So almost, you can enjoy 10 kilometers of, the skating. So literally, some people can actually commute, by skating, to the work. So Ottawa is a very nice city for the skating, but also very wonderful, the city for the immunotherapy. So it's capital of the Canada. So we have the many government institute such as National Research Council of Canada, Health Canada, or the Ottawa Institute, Hospital Research Institute. So many of the government institutes is located in the Ottawa. And then our university, University of Ottawa, the Faculty of Medicine, they develop the Advanced Medical Research Center to provide the research hub for the cellular immunotherapy. So here we are conducting the basic science, translational science, and then also clinical research as well. So our the Faculty of Medicine has ample experience in the cell therapy, including CAR T cell therapy. So our the collaborative research is our the current overarching goal is to combine our expertise. For example, University of Ottawa and then the Canada National Research Council of Canada has the CAR-NK expertise and then also NK cell manufacturing expertise. And then also Dr. Zhang has the mRNA-based CAR expression technology, so which we want to combine this technology to develop rapid, safe, and scalable CAR-NK cell production. So when we talk about the CAR therapy, we usually start with the CAR T cell breakthrough, so the CAR T cell immunotherapy. So as you can see, the CAR, the receptor is a completely synthetic receptor. This is kind of more Frankenstein version of a receptor. So those receptors never exist in nature. So when people generate those the synthetic receptor to take the antigen binding domain from antibody, and then also transmembrane domain, signal transduction domain from the T cell receptor complex, actually that can induce the very robust T cell activation. So the those CAR T-cell, the technologies really evolve, through the first generation, second generation, third generation. Maybe now it's the fourth generation, and then we can induce more like a robust, the T-cell activation, often recognizing the tumor antigen, which lead to the CAR T-cell breakthrough. So she's the Emily Whitehead. She's a really iconic the patient who first, the received, the CAR T-cell therapy. So she was, suffered from the very the severe the leukemia, and then now after CAR T-cell therapy, she's still cancer-free. So this is really the so efficient, the immunotherapy. The CAR T-cell has a really great efficacy, but also has some limitation. The CAR T-cell has some induced, some severe side effects, but the more important thing is that the CAR T-cell therapy only given in autologous setting. It means, the CAR T-cell, those, the CAR T-cell usually made from the patient blood, and then in vitro ex vivo manipulation, and then those, the CAR T-cell, the product is returned to the patient. So imagine the cell therapy is a very expensive therapy. When you generate those cell product in GMP grade, this is a very, very expensive process. So that is why those in the autologous setting really impose the very high a price tag on this the effective immunotherapy. When people try to use CAR T-cell therapy to the genetically irrelevant the patient, then those CAR T-cell induce very severe GVHD, so graft versus host disease. So the transport T-cell attacking the healthy cell in donor. So that is really the very, very detrimental consequence. So typically, almost impossible to do that. So most of a CAR T-cell is only given in the autologous setting, and then we try to address those limitations by developing allogeneic cell therapy. So allogeneic cell therapy, simply the concept is you get the cell from the healthy donor, and then you engineer ex vivo, and then expand, and you can generate many doses of the cell therapy product. So this is truly off-the-shelf cell therapy, because whenever a patient need those the therapy, just the sow one the cell product and then inject and then treat the patient. So for allogeneic cell therapy, NK cell has been really the most ideal cell population, because NK cell does not induce GVHD, and then also has a very low side effect. And then in the same time, they still hold a very strong anti-tumor activity. That's why NK cell hold the really great potential for off-the-shelf therapy. So using those allogeneic the NK cell therapy, we try to develop more efficient robust the immunotherapy by using the antibody. So when we use antibody and NK cell together, we can induce ADCC, so-called antibody-dependent cell cytotoxicity. Also, our laboratory tried to edit gene by CRISPR-Cas9 technology to develop the more the efficient NK cell the product. And then today I'm going to talk about more the CAR NK, so which we can express the CAR molecule on the NK cell. So for the CAR NK therapy, we have been collaborating with the National Research Council of Canada. So the NRC, National Research Council of Canada, NRC has a lasting expertise to generate those antibody by immunizing llama. So usually antibody, we know most of mammal has the heavy chain, light chain, and then antigen recognition is mediated by the barrier region of heavy chain, light chain. But those llama has a very unique antibody, which heavy chain only antibody. But still, they use only heavy chain. But still, those the antibodies show very nice, the specificity and then very strong affinity. So when the NRC generate those single domain antibody, and then we use those the antigen binding domain to generate the CAR construct, and then we express on the NK cell, and then we test their anti-tumor activity in vitro and in vivo. So there is many type of cancer, but today I'm going to focus on showing our result from the CAR-NK therapy against multiple myeloma. So this is the most of data generated by Shelby Kazmarek, my PhD student, and then also we have the very important have like a clinical collaboration. So the Elsa Bisram, she is a myeloma doctor at the Ottawa Hospital. So multiple myeloma is a cancer type. It's really growing in the bone marrow. So bone marrow, like usually we have a cell called plasma cell. The plasma cell is terminally the differentiate the B cell, which is secreting the antibody. So due to those the antibody actually protecting us from the many different diseases, including many different pathogens. But multiple myeloma, those plasma cells somehow got crazy, and then only they are growing, and then only they produce this useless antibody, so it doesn't help them much. And then the problem of multiple myeloma, we have like an immune system has two major cytotoxic cells, NK cell and T cell. Those cells really efficiently kill the cancer cell, right? But problem is the NK cell and T cell does not have like efficient the recognition system to recognize the multiple myeloma. That is kind of problem. So that's why we need to like express artificial receptor which can efficiently recognize multiple myeloma. So there is already two FDA approved CAR T cell therapy. So the Abemma and Carvykti. And then both the CAR T cell therapy targeting BCMA, B cell maturation antigen, and then those BCMA antigen is expressed in the late stage of B cell, and then also highly expressed in the multiple myeloma cell. So those the CAR T cell, like BCMA CAR T cell against multiple myeloma is really the efficient. So it had been shown almost the 80 or 90% in a patient who received those CAR T cell therapy, their response, and then but still same limitation is left, remain, right? So still has a side effect. The most important thing is that it's only given autologous setting. So that is really to put a very high price tag, almost the $500,000 is to have like one shot of CAR T cell therapy. So that's why we really feel like the need for the allogeneic therapy. So if we can generate the allogeneic cell therapy, we can cost down by 100 or 1,000-fold. So which will be really increase accessibility to the many patients. The CAR T cell against multiple myeloma concept is very simple. So some cancer cells highly susceptible to the NK cell because the cancer cell has a ligand which can recognize by activating receptor of NK cell. So in that case, the NK cell can kill very well those the sensitive target cell. But the cancer cell like a multiple myeloma cell, they don't express the ligand for the NK cell activating receptor. So the NK cell cannot see those cancer cells, so not able to control. So in this case, we just overexpress CAR-NK, CAR receptor, which can efficiently recognize the multiple myeloma cell, which we expect they can do very well, like the anti-tumor activity. So this is the first, the experiment, first in vivo experiment we performed, and then we try, really several years of, the trial or effort. Now we are able to engineer NK cell to express, CAR expression. So as you can see, almost, 70%, 75% of, NK cell express CAR molecule. So LAMP6, they don't express in a BCMA, so without CAR expression, their killing activity is almost identical. But the MM1S, this is the myeloma cell line, which express the high BCMA expression. So as you can see, you can really appreciate the huge the functional difference. So without CAR, there almost no killing, but with CAR, there is really the almost 60-80% killing we can observe. But when we actually inject those NK cells in vivo, somehow those CAR NK cells can delay the myeloma cell growth in vivo, but it failed to induce the remission. So and then further study we showed really the problem is actually NK cell persistent. So we inject NK cell in vivo, but that NK cell actually does not last long enough. So after 3 weeks, when we measure the NK cell, like almost 0.5%, very, very low number, the proportion of NK cell is found. So to address those NK cell persistent in vivo, we decide to co-express IL-15. So IL-15 is cytokine, which is a very critical cytokine for the NK cell survival and then proliferation. So when we co-express IL-15 and then CAR, and then this is the without IL-15, the media without IL-2, almost all cells found that at day 5, the culture, but when the CAR NK cell express IL-15, they are really protect from the cell death. So which is really the IL-15 expression is working. And then when we look at the in vivo, when we look at the in vitro, the killing activity, as you can see, without NK cell, the tumor cells, show uncontrolled growth. But without IL-15 and then with IL-15, actually they can control the cancer cell growth in vitro very well. So more importantly, even when we measure the NK cell frequency in blood, after 1 month later, still we can see very nice, the stable population of CAR NK cell we inject. So we are kind of very excited about. So this is a very nice, the very important experiment. So as you can see, without any treatment, it really the myeloma cell showed uncontrolled, the growth. And then when we inject, unmodified NK cell, actually we don't see almost no difference between the untreated and then mice treated with the unmodified NK cell. That really highlight, the recognition system is missing on NK cell. So when we test, the NK cell express IL-15, there's no difference. And then when we inject a CAR NK, actually they can delay myeloma cell growth, but still it failed to induce the remission. But when we treat the mice with a CAR NK cell expressing IL-15, actually we can show a very long remission. So which is really the demonstrate the strong anti-tumor activity. So this is a quantification of those the tumor cell growth. And then the body weight, actually, as you can see, the mice receive NK cell expressing IL-15. Almost, they don't lose any body weight. And then the HLP, hip leg paralysis, that is the pathology of multiple myeloma. And then you don't see any the multiple myeloma related the pathology in these mice. When we look at the cell, in the blood and then spleen and bone marrow, definitively IL-15, the expression can induce the longer NK cell persistence. So as you can see, whenever the mice has the cell expressing IL-15, they have a more NK cell population was found. More importantly, myeloma cell. The myeloma cell also found in the blood and spleen, but the most importantly, bone marrow. You can see all the mice, that has a very, very the uncontrolled, growth of a myeloma, except the mice receive CAR NK cell expressing IL-15. So almost no myeloma cell found in the bone marrow. So we are very excited, with this, the promising data. So we can generate a really potent CAR NK cell. But those data is all those NK cells made by lentiviral transduction. Okay, so lentiviral transduction. This is the most well-known, well-established process to the engineer NK cell and T cell. So for example, there is 7 CAR T cell therapy available, and then all use lentiviral or retroviral transduction to generate the CAR T cell. However, those the lentiviral transduction method has a complicated and then the very expensive process. So it's actually to generate those the lentiviral vector, it take the 10 or 14 days. So very laborious the process. And then to generate those GMP grade of virus is very, very expensive process. So since the process itself is complicated, there is a more regulatory hurdle, and then which limit also scalability. But our collaborator, Dr. Zhang, she actually established mRNA-based the CAR NK technology. In this case, the mRNA is made by in vitro transcription and then delivered to NK cell by electroporation. So total the process time is we can shorten 1 or 2 days. So this technology is really enable NK cell engineering the fast and then flexible and then cost effective. So it's it's more like a fast robust the process. That's why it's less clinical validation is required. Only caveat of this technology is transient CAR expression, and then thereby transient in vivo efficacy. So by collaborating with Dr. Zhang, we established those mRNA-based CAR NK cell technology in my lab at University of Ottawa. So this is the in vitro transcription, the data. So there is an RNA band clear. And when we use those the RNA molecule, when we transduce, so we have a different dose of the mRNA. This is we are using the linear mRNA. And then as you can see, day 1 we have almost 100% the NK cell expressed very good the level of CAR expression. But problem is that the expression is continuously losing, and then the day 7 almost expression is returned to the basal level. That is really one caveat. So to address this the caveat, and then to actually induce more long-term expression of a CAR, Dr. Zhang is using the circular RNA molecule. So circular RNA molecule is compared to the mRNA molecule is much stable half-life and then low immunogenicity. So she hypothesized if we deliver the circular RNA, we can extend in a CAR expression. Definitely she was right. So when you look at the day 1, day 3, day 5, those linear mRNA, the NK cell, the receiving of linear mRNA, they are rapidly lose the CAR expression. So day 5, almost the all expression is returned to the basal level. But when she used circular mRNA, as you can see, even day 5, we have a very nice CAR expression remaining. So the bottom, the graph is actually kinetics of the CAR expression between the linear mRNA and then circular mRNAs. So we are collaborating with Dr. Zhang for those mRNA, the CAR NK product in the project, and then we already established the process development for the GMP grade CAR NK manufacturing. So for the CAR NK manufacturing, we are using the G-Rex, the system. The G-Rex is the gas permeable rapid cell expansion. So what makes a G-Rex, the system is special. They have like the bottom, any plate, the bottom is the gas permeable membrane. So which induce the convection of the gas and the nutrient. That really minimize media change, and then it will really induce the lower cost and then very simple workflow. And then also is they have many different size of those plate, and then they also have a GMP version of a plate, so the GMP compatible. So to do like an in vivo study, even the xenograft model, we need to have a lot of NK cells. So we have to sometimes grow this many the flask. This is roughly more than 50 flask. And then now using the G-Rex, actually it can be replaced by a single G-Rex in the place. So G-Rex system is really efficient to generate many or expand many the NK cell. And then it's really scalable because the G-Rex plates, some has a 2 centimeter bottom and then also 100 centimeter, or as big as the 500 centimeters. So you can really generate many NK cells using this system. So using those G-Rex the system, we develop the process the development, and then we use the NK cell from the PBMC and then cord blood cell, and then we isolate the NK cell and then we expand. So as you can see in the second panel from the top, you can see how like our naive NK cell, like the morphology is very tiny, right? But when we expand and stimulate and expand, and then NK cell, the size is really big. So this is really the characteristic of very active or cytotoxic NK cell. So those NK cells really kill the cancer cell very well. And then also we were able to introduce the CAR expression by, in this case, lentiviral, but hopefully we want to replace those the CAR expression procedure by the mRNA CAR technology, actually the developed by the Dr. Zhang. So this is a hypothetical number, but still is very conservative number because we don't pick the really super donor, right? This is just we take a random donor, and then still this is the number what usually we achieved. So we receive 120 ml of blood, and then through the third time of expansion the round, we can generate 2,000 billion of the NK cell. So we kind of expect the 10 million per patient, per each dose. So through the dose, the process, we can expect we can generate 200 dose, from the one blood donor, which is really great. So and then also our NK cell also tested, with a different format of the CAR NK. So the bispecific killer engager, the BiKE. This is the kind of antibody-based, the therapy. So in this case, NK cell and then target cell. So the target cell is multiple myeloma, as I mentioned. NK cell, they don't have an efficient, the recognition system of, the myeloma cell. In this case, when we treat, the BiKE, actually BiKE molecule connect, NK cell and target cell together, and then induce, the very robust, the NK cell killing of target cell. So we use our cell combined with those spike molecule. As you can see, it really works very well to induce the anti-tumor activity. So all the data demonstrate we can really generate the superior cytotoxicity of NK cell, so NK cell with a superior cytotoxicity using our manufacturing pipeline. So using this, the very important, collaborative, the study, and then we joined research program. We want to combine both, the expertise. So the University of Ottawa and then National Research Council of Canada, we established NK cell manufacturing the pipeline, so we can generate the NK cell in large number with a very potent cytotoxicity. And Dr. Zhang actually has the mRNA CAR technology, so that she can really make the CAR expression is robust, easy, fast. And then hopefully we can generate the mRNA CAR NK cell, like an off-the-shelf, the cell therapy product, and then can be commercialized our partner UCI Therapeutic. So for that, we already achieved several milestones. I think the key milestone is long-term the deployment of Dr. Zhang. Actually, Dr. Zhang the visit in University of Ottawa and stayed the 6 months to establish those mRNA CAR NK project. She also, as you can see, the woman has a cap. Actually, she's Dr. Zhang, actually showing how the mRNA CAR NK works. And then she attended all the lab meetings. So this truly the collaboration. And then also we opened the Global Joint Research Center. So this is the center, the plague, we attach plague. And then also we host several a symposium or consortium and workshop to discuss about the future of the NK cell therapy, including CAR NK. So the first, the symposium was an inaugural symposium among KISS, UCI Ottawa Joint Symposium, which happened in April 2025. So we have hosted the 5 the symposium so far, and then the timeline. So the first 6 months we established GREX system to generate the massive the large scale of NK cell expansion. And then also we finish cloning for the mRNA CAR project. Currently we are trying to do functional validation of those mRNA CAR NK products. So we fully established mRNA CAR NK cell, and then now we are testing those the product in vitro, in vivo. So hopefully 2026 January we want to integrate those optimized manufacturing the pipeline with the mRNA CAR NK production. So hopefully we can scale up using the GREX system to the manufacturing. So I want to really acknowledge my member at University of Ottawa, and then people in NRC, and then also Dr. Zhang at KIST, and then UCI Therapeutic, and then our clinical collaborator, Elisa Bisram. And then I want to give the special thanks to the NRF to support our study. Thanks so much.
Yeah, it's a wonderful presentation, especially mentioning advantage of NK cell therapeutics in terms of allogeneic production, which might be available for broad patients to treat diverse diseases. So we are moving on to a Q&A section. I will take any questions, please. I have one question regarding NK cell therapeutics. As you know, one of the advantages of NK cell in terms of allogeneic production we can expand NK cell from human variant sources, including peripheral blood, cord blood, and iPSC. So what do you think which cell type would be beneficial for translating in clinic?
Yeah, so there is a hypothesis, the maybe cord blood NK cell, they are less, the kind of the developed, so they are maybe less, the chance to induce, GVHD. But the people historically, NK cell really has been known for the safe cell. So it's really we can, back to almost 20 years, people start to inject, many number of NK cell, and then almost no GVHDs observed. So the NK cell has an activating receptor, inhibitory receptor. Maybe inhibitory receptor help, prevent, GVHD. But the most important thing is the previous, many clinical trial demonstrates the safety of NK cell in allogeneic setting.
Okay, thank you. Yeah, please.
Nice talk. Yes, I would like to, questions. I don't know exactly about yours NK cells. You use lentiviral and versus the circular mRNA techniques. So in your slide, yes, but even though the circular mRNA is good stability and cheap and very nice, but from day 7, you're somewhat decreasing the tendency when I don't know exactly how long is the circular mRNA we have to pursue the activities versus the lentivirals. Yeah.
So we haven't tried the CAR-NK cell made with the circular mRNA. So still I don't have an answer yet. But what I can say is even we use we engineer NK cell using lentiviral transduction. Those CAR expression is rapidly down modulate once we inject to the mice in vivo. So I would say this the CAR expression is we measure, and then roughly after 1 week, all the CAR expression is down modulate. So the completely CAR expression is gone. But as you can see, our in vivo data really demonstrate really the CAR-NK cell is working. So I can kind of the question differently maybe how many day window is required for the efficient CAR-NK efficacy. I don't think we need many days, maybe a couple of days, 2 or 3 days, and then NK cell make a very big difference. So they are very, very good at the debulking of the cancer cell in vivo.
I have one opinion regarding your question. Actually, mRNA still has a limitation in terms of long-term persistence, but circular mRNA I have been used in my project is non-modified circular RNA. So if there are several components, we can consider to increase persistence by engineering IRS sequence or pseudotyped nucleotide, something like this. Maybe maybe someday we can optimize mRNA with long-term persistence. Thank you so much. So I think I don't think we have more questions. So we are close to this section. Thank you for Professor Lee. Yeah.
Thank you so much.
Thanks once again, and please give another big round of applause to our moderator and speaker for wonderful presentation and discussion. Now we'll move on to the second presentation of the second session under the same theme, biotechnology-based API production. And this session will be led by Professor Young-Sup Yoon from Yonsei University. And please welcome our moderator with a big round of applause.
Good morning, everybody. Welcome to this JCR Hub presentation. I'm from Yonsei University, working as a professor. My majors are including stem cell biology and research, and also clinical translation of cell-based therapy for cardiovascular regeneration. So far, we have been developing the Korea-US Joint Research Center aimed at developing next-generation cell therapeutics for cardiovascular disease, and through this center to develop directly reprogrammed cardiovascular disease tissue. We have two aims. One is to develop methods for generating directly reprogrammed cardiovascular tissue, which is the world's first one for the treatment of cardiovascular disease. Another aim is to establish an international collaborative center for the development of directly reprogrammed tissue therapeutics, thereby producing scientific and technological achievements as well as outcomes and international cooperation, such as personal exchange and joint research. With the collaborators in the US, we have been so far achieving several important milestones. One of them is to publish world's first directly reprogrammed smooth cells. We also have been continuously developing cardiovascular tissue with this directly reprogrammed technology, including cardiomyocytes and endothelial cells. Our plan is not to stay with just biomedical research. We plan on translating our research into industrial utilization as well as biomedical and academic utilization. Particularly, we are pursuing to develop world's first innovative directly reprogrammed cell center. We started this technology about 13 years ago. So far, we have the largest array of patents in this specific area. Given the high prevalence of cardiovascular disease, successful commercialization of these technologies will meet a vast market demand. Furthermore, this technology can be expanded into various formats such as organoids. Vascular cells are the most important cells for cell survival. So this can be used not only cardiovascular, but many other diseases, including stroke, wound healing, diabetic complication, liver regeneration, pancreatic cell regeneration, and everywhere. This can also be used organ on a chip system, providing platform for other disease treatment and drug evaluation. So we have a huge application side in this technology, and we're very happy to have our center to fund us to get this far. We really appreciate the government and the Korean R&D Center to give us an opportunity to come this far. Today, I'm very happy to introduce Professor Sang-Ho Lee, who will present about the recent advancements in direct reprogramming technology. In terms of so-called cell-based therapy, there have been 3 stages. Number 1, adult stem or progenitor cells. Number 2, pluripotent stem cell, which include the embryonic stem cells and induced pluripotent stem cells. Now we're further going to the 3rd generation technology, which is direct reprogramming technology. So we have been so far in competing with any other worldwide scientists and engineers in this particular section. Dr. Sang-Ho Lee is one of the leaders in endothelial cell direct reprogramming technology. He completed his undergrad and master's degree in Korea University, and also he pursued his PhD at the University of California, Davis, and he did his postdoctoral fellowship at UC Berkeley and Emory University, and thereafter he became faculty at Emory University and has been pioneering in developing direct reprogramming, particularly focusing on vascular regeneration. He's unable to come because his wife has some issue. So he will give us presentation with a recorded clip. Please welcome Professor Sang-Ho Lee. Thank you.
Everyone. I'm Sang-Ho Lee, working in Division of Cardiology of Emory University. First of all, I sincerely apologize for not being with you and for delivering this presentation by pre-recorded video. And thanks to the organizer for inviting me as a speaker for this symposium. Today, I will talk about advances and perspectives of direct reprogramming, focusing on cardiovascular regeneration. Cardiovascular diseases, CVD, remains a leading cause of death globally, responsible for more than 17 million deaths each year, and accounting for about 30% of all deaths. Among these, ischemic CVD accounts for a significant proportion, causing approximately 9 million deaths worldwide. Ischemic cardiovascular diseases such as coronary artery disease, CAD, such as MI, and peripheral artery disease, PAD, such as chronic limb-threatening ischemia, are caused by vascular occlusion and reduced blood perfusion. To treat these ischemic CVD, to date, there are three broad approaches to treat ischemic CVDs, namely medicinal, interventional, and surgical approaches. Medicinal therapy is typically the first line of treatment. These include pharmacological interventions and lifestyle changes. It is non-invasive and primarily aimed at stable disease management, managing symptoms, and preventing progressions. For patients who require more targeted treatment, interventional therapy provides a minimally invasive option. Procedures like angioplasty, stent placement are commonly used to restore blood flow in localized area of arterial narrowing. While effective for localized regions, these interventions often fail to address issues at a microvascular level and carry risks of possible blood clot formation. In more advanced or complex cases, surgical interventions may become necessary, including coronary artery bypass grafting CABG. These are more invasive but has shown effectiveness with long-term survival benefits. However, they are associated with increased perioperative risks and potential complications. Despite advances in these therapies, a subset of patients, often referred to as no-option patients, continues to experience poor clinical outcomes. These individuals present with advanced or diffuse disease that is unresponsive to conventional treatment, leaving them with a few options beyond high-risk interventions such as cardiac transplantation or limb amputation. It is because these current therapies are unable to regenerate lost myocardium or vasculature in CVD patients. To address these needs, biological therapies, including gene and cell therapies, have emerged as promising alternatives, as the pathophysiological cause lies in the loss or dysfunction of blood vessels. These strategies aim to induce therapeutic neovascularization, support proper blood perfusion. Among the biological therapy options, cell therapy has been considered as an attractive strategy in ischemic CVDs, as it supplies vascular components, especially endothelial cells, for the generation of new functional blood vessels, and cardiomyocyte to restore cardiac function. So more focusing on the cell therapy, I'll address how we can get cells for cell therapy. Adult stem cells has shown limited therapeutic effects, and embryonic stem cells has ethical issue. Then researchers found alternative strategy. I explain it using this Waddington landscape. Since induced pluripotent stem cells have been introduced by reprogramming or conversion of somatic cells into embryonic stem cell-like cells with pluripotency. So these cells can be differentiated into specific cell types for cell therapy. Moreover, cells can be made directly from another type of somatic cells without going through pluripotency. Since iPS-C induced pluripotent stem cell is reprogrammed from the somatic cells from self through the process called cellular reprogramming by overexpression of pluripotency transcription factors. So iPS-C opened the new phase of autologous cell therapy for regenerative medicine. However, iPS-C needs to be differentiated to specific target cells for cell therapy. With this method, going through pluripotent cell states, we can produce target cells on a large scale. However, it has several things and risks to consider: genomic instability, tumorigenic and aberrant tissue formation risk. To overcome these problems, the same strategy was applied to the process called direct reprogramming, in which somatic cells are converted to another type of target somatic cells by overexpression of lineage-specific transcription factors, ETB2 for endothelial cells and myocardin for smooth muscle cells, GATA4, MAP2C, TBX5, and HAND2 for cardiomyocytes, and BRN2, MIT1L, ACSL1 for neuron. This direct reprogramming method bypasses pluripotent cell states, therefore reduce tumorigenic risks. And it is faster and has more efficient turnaround time to get target reprogrammed cells. However, the efficiency is largely depending upon a proliferative states of source cells. Among many cell types, including neurons generated by the direct reprogramming strategy, I'll focus on these three cell types: endothelial cells and smooth muscle cells and cardiomyocytes for the therapy for cardiovascular diseases. To treat ischemic cardiovascular diseases, PAD and CAD, we need to generate new functional blood vessels, so-called neovascularization, by supplying vascular components, especially endothelial cells. It facilitates capillary regeneration. Moreover, in case of larger vessels, artery and vein, which are composed of endothelial cells, extracellular matrix, and smooth muscle cells, we need additionally to supply smooth muscle cells to regenerate such larger vessels and to stabilize newly formed blood vessels. And for CAD, to restore cardiac function, we need cardiomyocytes. So we tackled first endothelial cells. We successfully reported in 2017 direct reprogramming of human dermal fibroblasts into endothelial cells using endothelial lineage specific transcription factor and a pioneering factor ETB2. For this study, we tested 7 transcription factors for endothelial lineage specification and found ETB2 alone was sufficient to reprogram HDF into endothelial cells. Using doxycycline-inducible lentiviral ETB2, we generated EC-like cells referred as early RECs, and with longer culture with valproic acid BPA and reinduction of ETB2, we generated the late RECs with more mature phenotype. With ETB2 overexpression with doxycycline, we could observe morphological changes similar to endothelial cells. Among these cells, 40% expressed KDR and 50% expressed CDH5, canonical EC markers. Around day 7 after ETB2 transduction, we collected KDR positive cells and called REC, reprogrammed endothelial cells. When these RECs were transplanted into the mouse hind limb with ischemia, an animal model of PAD, we could observe blood flow recovery in red compared to the other control like PBS, HUVEC, or source cell HDF. Also, REC shows protective effect from the limb loss, and in histology analysis of the hind limb tissue of REC transplanted animals, we found transplanted RECs in red incorporated into blood vessels in green and participated in the formation of new blood vessels where the arrow pointed. So these results were appreciated as a cover image of the journal. In summary of this study, to generate early RECs, ETB2 overexpression is sufficient. Although they are immature, they showed EC characteristics and therapeutic and vessel forming potential. So early REC can be applied for autologous cell therapy, whereas longer culture with ETB2 reintroduction supplemented with BPA matured early REC to late RECs. However, due to the extended duration of the culture, late REC will be more useful for drug screening or disease mechanism studies rather than cell therapy. One thing I'd like to point out is that for this study, we used lentiviral vector. So not only our study, but also other early direct reprogramming studies used retroviral or lentiviral vectors to deliver reprogrammed transcription vectors for overexpression. Such retro or lentiviral vectors are integrated into host genome, which makes cells virtually clinically incompatible. Therefore, as a next step, we try to make REC clinically more compatible using adenoviral vector. So using adenoviral ETB2, we could generate RECs in a similar time frame as we succeeded with lentiviral vector. And these RECs also displayed EC characteristics in vitro and in vivo. And when they were transplanted into mice with hind limb ischemia, they showed therapeutic potential and participated in vessel formation by placing perivascularly or incorporating into vasculature. And these data has been pre-printed in bioRxiv. As a next wave, we generated smooth muscle cells by direct reprogramming. This study was published this year as a successful direct reprogramming of adult somatic cells into contractile smooth muscle cells with vascular regeneration potential. In this study, we transduced HDF with retroviral myocardium supplemented with all trans-retinoic acid to produce reprogrammed SMC RSMC. RNA-seq analysis showed that contractile SMC gene transcript was substantially higher in RSMCs compared to myocardium-only transduced HDF. And compared to myocardium-treated HDF, RSMC has more prominently exhibited those markers showing typical non-triated SMC-like patterns. When we measured carbacle-induced intracellular calcium release in a single cell over time, myocardium-only transduced HDFs and human aortic smooth muscle cells often had a large primary peak, which was followed by a low number of recurrent peaks. However, all reprogrammed SMCs produces more robust primary peaks and multiple submaximal calcium release in the form of propagated waves over extended period of time. Such vasoconstrictor-induced propagated calcium release suggested strong contractile properties. Transplantation of RSMCs into ischemic hind limb muscles improved vascular reperfusion and protected from limb loss by necrosis and promoted recovery from tissue ischemia. The in vivo engrafted RSMCs contributed to the vessel formation through their role as parasites and vascular smooth muscle cells in a small to large blood vessels. Another type of cells is cardiomyocytes. We have not done cardiomyocyte direct reprogramming, so here I will briefly introduce few important cardiomyocyte reprogramming studies. In 2010, Srivastava group first reported cardiomyocyte reprogramming using mouse fibroblast. And in 2012, Bixazao's group uses microRNAs to directly reprogram fibroblasts into cardiomyocytes. In 2013, Olsen group showed first study of human cardiomyocyte reprogramming. In 2018, Ieda group uses Sendai virus to directly reprogram human cardiomyocytes. So in these landmark studies, directly reprogrammed or induced cardiomyocyte showed improved heart function after injury in animal models. So these direct reprogramming method and techniques has been used not only to generate target cells but also for in vivo or in situ direct reprogramming. In 2012, two groups, Srivastava group and Olsen group, published in vivo direct reprogramming in mice using the same transcription factors for cardiomyocyte direct reprogramming. So they aim to directly reprogram resident or induced cardiac fibroblast during ischemia. Later, Cho group used modified RNA of TBEX18 to produce not cardiomyocyte but pacemaker cells. And ETB2 has been used to facilitate differentiation into endothelial cells from iPSCs. So we collaborated with In-hyun Park at Yale to enhance perfusible vasculature in human brain organoids by temporal induction of ETB2 in iPSCs. Another group also used modified mRNA of ETB2 to facilitate differentiation of iPSC into endothelial cells. And recently we generated not a single type cells but all three type of cells together by direct reprogramming of mouse tail tip fibroblast. And in this study, we named it cardiovascular tissue, CVT. Transfecting mouse tail tip fibroblast with microRNA 208B supplemented with ascorbic acid and BMP4 in the reprogrammed medium generated reprogrammed cardiomyocyte RCM and RECs and RSMC together. So collectively we call this cocktail MAB. MAB cocktail could generate not only functional cardiac-like cells but also forming three-dimensional 3D tissue-like structures. We refer to reprogrammed cardiovascular tissue, RCVT. This RCVT contains reprogrammed cardiomyocytes, endothelial cells, and smooth muscle cells. And LCMS analysis of RCVT also showed approximately 51% of the matrix consists of collagen isoforms with collagen type 1 and proallergen, a collagen type 1 binding protein. Implantation of the RCVT into infarcted hearts of mice led to preservation of myocardium and the migration of reprogrammed cells to the infarcted area, improving cardiac function. So from these previous studies, we could know that the traditional stem cell based therapy can be slow, costly, and may carry tumorigenic risks. However, direct reprogramming is faster and potentially safer. And it allows in vivo reprogramming, which will be a simpler translational approach compared to the cell therapy. And it may allow targeted renewal of damaged myocardium, attenuated fibrosis, and restore contractility in the heart. Despite of such advantages of direct reprogramming, there are challenges and things to consider in the future. First point is cells. The source cells should be easily available through minimally invasive procedure, and they should be proliferative. Also, we need to evaluate reprogrammed cells for their safety, immunogenicity, and long-term stability for clinical application. The second point is efficiency. The reprogramming efficiency is still low due to the refractory population, which poorly maintains its phenotype and poor proliferation. Also, it lacks a robust, reliable method. When these cells transplanted, the cell survival is still challenging, and the risk of arrhythmia, especially for cardiomyocyte transplantation. The third point is factor delivery method. It should be clinically compatible and less immunogenic. So, adenovirus and AAV has been considered as a viral vectors. For non-viral method, modified RNA has been demonstrated for its feasibility in some cases. For in vivo direct reprogramming study, nanoparticles would be a great candidate as delivery tool, but we have to be cautious about the off-target effects. The fourth point is reprogramming factors. Of course, we can consider the combination of multiple transcription factors, but also microRNA would be a great reprogramming factors. Small molecules and cell signaling molecules can be supplemented to enhance direct reprogramming. And epigenetic and metabolic modulators are considerable. So these factors will be discovered or emerged from the mechanistic study of direct reprogramming processes. So in summary, direct reprogramming is rapidly advancing with transformative potential for cell therapy, as well as tissue engineering and disease modeling. Several technical and translational challenges remain but are actively being addressed. Therefore, through direct reprogramming studies, we can standardize the protocols to prepare source cells from patients. We can develop various reprogramming factors in clinically compatible forms. Using these factors, we can generate each cell type or in a form of tissue. And cells and mimetic tissues can be utilized for the treatment of PAD and CAD patients, and the reprogramming factors can be directly used for in vivo direct reprogramming. Moreover, these cells and reprogramming factors can be used to facilitate organoid formation and maturation for further studies. I'd like to thank my lab members and Dr. Young-Sub Yoon and his lab members as well, and my longtime collaborators. Thank you so much for your attention.
Dr. Lee is not available for answering questions, but if you have any questions, I can answer for him if I can. Okay, Dr. Zhang?
I have one question about in vivo directed reprogramming. He has used nanoparticles or virus to deliver genes in vivo. I know in my thinking, one of the most challenge in vivo directed reprogramming is to deliver genes specifically. So what would you think the most efficient way to deliver genes specifically?
So can I reiterate your question? So what is the most efficient method of delivering genes?
Yeah, there are several ways to deliver genes, such as nanoparticles, AAV, or any other things. But depending on modality, they have still limitations. So what do you think the most efficient modality to deliver gene by reducing off-the-target effects?
Yes, that's a very challenging question. I think nobody may answer that precisely, but from my own experience and my own knowledge, adenovirus may be the most efficient in terms of the transfection efficiency. However, they may sustain there for relatively short compared to other insertional technologies. And also AAV is widely used. However, recently I recognized that many of the AAV companies closed their programs because there are unexpected side effects and immunological reactions, etc. Of course, I don't think many people tried it, but Sendai virus may be a good choice, but it's quite robustly protected by Japanese company. So that is a very challenging question. So that's why a ton of people have been developing modified mRNA and also the nano delivery technologies, as you mentioned. If we say our own perspective, we still believe adenovirus may be a choice of a delivery tool because from our COVID vaccination, adenovirus is one of the least side effect provoking agents, still effective. We're not sure, and I haven't seen any specific papers addressing comparing different delivery tools, but that's what we think.
And I have one more quick question regarding ex vivo reprogramming. Is it only for autologous therapeutics, or we can avail for allogenic production?
Yeah, that's a very good question. So why some people are developing autologous, why the other people are developing allogeneic? It's a matter of economical utility. Definitely autologous is much better than allogeneic. However, the production cost is very high. So if somebody can develop so-called universal cell sources, and as you may know, there are at least 20 different groups who have been developing universal iPS cells. In the direct reprogramming tissue technology, if anybody can develop, for example, fibroblasts that are universal, so that knocking down HLA-1, 2, and some other epitopes, that could be widely used. So we've been waiting, and then we're also working with iPS cells, and there have been so many people so far. Clinically compatible universal iPS cells are not yet available, so we'll see what will happen down the 5-10 years.
Thank you.
Thanks once again for your wonderful presentation and discussions. Please give another big round of applause to our speaker and our moderator. Now we will take a short 15-minute coffee break and networking time. Please enjoy the refreshments provided at the back of the conference hall. And please feel free to enjoy various programs prepared at the lobby. And we hope to see you again and see you soon. 네, 저희 커피 브레이크 시간 때 잠시 더 안내 말씀드리겠습니다.
May I have your attention, please? I understand that you got your name card yesterday, but today you have to get reissued your name card. Please go to the information desk and get your name card once again. For better, um, for better event, we are asking for your feedback and opinions.
Ladies and gentlemen, may I have your attention, please? The last presentation of the session 3 will begin shortly. We cordially ask all participants to enter the hall and be seated. Today's symposium will be conducted in English with simultaneous interpretation service, so please feel free to use a receiver at the back of the conference hall or on your desk. It will be channel 1 for Korean and channel 2 for English. And in the Square Ballroom lobby, you can enjoy various programs, including regional achievement exhibitions, a vision promotion video, and a photo zone. We kindly encourage all participants to take interest and join in. To ensure the smooth progress of the event, please check once again if you set your mobile phone to a silent mode. And please note that today's presentation materials are provided with ebook format via QR code. You can scan the QR code to download today's presentation materials. Our last presentation for session 3 will begin soon. See you soon. Ladies and gentlemen, distinguished guests, welcome back, and we will move on to the last presentation of the session 3. We'll move on to the second theme of session 3. It's Biofoundry, and I'd like to invite our moderator. It's Dr. Seung-Go Lee from KRIBB. Please welcome him with a big round of applause.
Good afternoon, everyone. My name is Seung-Go Lee, Director of the National Biofoundry Project in CRIB. Our goal in National Biofoundry Project is to build a world-class public biofoundry platform by 2029. This platform will bring together expert teams and about 10 optimized workflows in each stage of the DBTL cycle: design, build, test, and learn, creating a globally connected infrastructure for synthetic biology and biomanufacturing innovation. As Dr. Dae Lee yesterday mentioned, the CRIB Biofoundry Beta is serving as a pilot platform for the National Biofoundry Project. While our collaboration with the UK focuses more on policy and national strategies, our work with the United States centers on learning and applying the operational experience from the leading biofoundries of US. Our key US partners include the Agile Biofoundry at Lawrence Berkeley National Lab, supported by the US DOE over the past 9 years, and the Illinois Biofoundry at the University of Illinois Urbana-Champaign. So I'm very happy to introduce today's speaker, Professor Yong-Soo Jin from the University of Illinois Urbana-Champaign. Professor Jin has played a central role in the DOE research program, and he is widely recognized for pioneering work that enabled yeast to efficiently use xylose, an important sugar making up about 20% of biomass, yet once difficult for yeast to metabolize. He has also trained and mentored many young scientists, including over 17 Korean PIs who now lead their own research groups in Korea. Please join me to welcome Professor Yong-Soo Jin.
Thank you. Thank you for the invitation to speak and also kind introduction. It is my pleasure to give a presentation about this biofoundry-based microbial engineering for sustainable biomanufacturing. So I have been working in this area more than 20 years, and the key changes happening last 5 to 10 years is from the bench laboratory work-based microbial engineering to this biofoundry-based microbial engineering. As you hear today, we are making tremendous advances in using laboratory automation to make efficient and rapid strain engineering. So I'm going to talk about that today. So this is the outline of my presentation. The first, I'm going to talk about why we do this microbial engineering, which is for bioeconomy implementation. And then I'm going to talk about the concept of a biofoundry and application. And I'm going to introduce 3 biofoundry involved in this project. The first one is K-Biofoundry, and second is Agile Biofoundry, which is in Lawrence Berkeley National Lab, and third one is iBiofoundry, which is in the University of Illinois at Urbana-Champaign, where I am involved. And lastly, I'm going to talk about a detailed content of the research project and also our plan and future perspective. So as you know, this climate change is a big issue for the human society, and this resource depletion and waste accumulation and pollution is a big problem. And also we have this population growth and urbanization. That's why we need to think about new way of creating the economy, as this is called sustainable bioeconomy. Here is the illustration, which is a circular bioeconomy explanation. As you know, all this human industrial activity was to using fossil fuels or the chemical process to make goods and material and things. But now we need to think about how to create this circular bioeconomy. Instead of using fossil fuel, we need to be using this excessive or abundant substrates, such as the lignocellulosic biomass or the organic waste. And in particular, the focus of this project was to use methane gas from industry which is produced as an industrial waste as a substrate. And then we use this industrial waste as a substrate, and we produce like plastics and fuels and chemicals and other ingredients so that we can implement this circular bioeconomy. And one interesting substrate which has not been used a lot was this methane gases. As you know, methane is produced from industrial processes, and also many the biological process generate methane as a product. And there is a microorganism called methanotrope. As you see here, this methanotrope is already using methane as a substrate, but they make very little bioproduct. That's why the microorganism we have in the nature is not useful for the bioconversion. However, due to the thanks to the metabolic engineering kind of technology, we can reprogram or rewiring this metabolic pathway so that we can maximize the production of our target product using methane as a substrate. So this is the kind of emerging area of biomanufacturing. So this project will try to maximize the utilization of methane and to produce a higher value product. And then one of the key ideas is to use or establish and use biofoundry. So let me briefly introduce what is a biofoundry. Biofoundry is a core infrastructure that integrates artificial intelligence and robotics to automate and standardize design, build, and test launch cycle. So as you see in the illustration, we typically go through these 4 steps of a research cycle. First one is the design. So we need to design ideal biocatalyst, ideal cell, and then we need to build. But as you know, building is a very laborious and time-consuming step. I mean, I guess everybody here knows that, making a construct of a plasmid or making a transformation, these are all time-consuming process. So we want to automate this building process. And then also testing. When you construct a new strain or a new enzyme, you need to measure the activity, you need to characterize the performance, which is also time-consuming. So we think this build and test process can be automated using BioFoundry. And then we can use this new AI platform to learn about the system better. And then this learning cycle generate new knowledge for the new design so that we do this iterative cycle. Actually, this is what we are doing. As a biologist, we are doing this the last 15 years, 20 years, 40 years. But now using this BioFoundry, we can speed up this DBTS cycle so that we can achieve the goal more effectively. So the goal is to establish this global BioFoundry collaboration among participating BioFoundry in this project. So we have 3 goals. First one is to design the enzyme and metabolic pathway using AI. And then also we will develop the gene cell construction optimization kind of protocol. And then also we want to develop the culture and downstream processing to optimize the production of our products. So we have many participants in this project, like Seung-gu Lee is the leader of this project, and Professor Byung-Ha Oh, Professor Sun-won Kim, Korean participant. And also we have Nathan Hilson, Jay Kisling, and Nigel Moni, and myself, and Hyo-min Zhao, and Professor Dae-gyun Lo. These are the participants from foreign country. So I'm going to give you an idea about what is the BioFoundry and also give you an example of the work which has been achieved by BioFoundry by these 3 kind of BioFoundries. The first one is K-BioFoundry. Right now is a beta, but we soon to have a K-BioFoundry very soon. And second one is Agile BioFoundry in Lawrence Berkeley National Lab. And third one is iBioFoundry. We constructed this BioFoundry recently, so I'm going to show you how we constructed this BioFoundry and then how we are using this BioFoundry for the bioresearch. So the first one is this K-BioFoundry introduction. So this K-BioFoundry was initiated in 2019, and then 2021, CRIP invested this K-BioFoundry beta. So this is kind of a precursor of a new BioFoundry, which is going to be built in the next 5 years. So as you know, last year, Korean government announced this launch of the Korea BioFoundry project. So until 2029, this whole BioFoundry will be constructed. Even with this beta, K-BioFoundry beta, they made impressive achievement. So yeah, this is more explanation about the K or Korea BioFoundry, which is about near $100 million investment, so that they will build a national BioFoundry, which will serve various biotechnology research in this country. So this is one of the achievements by K-BioFoundry Beta, which is led by Dr. Ha-Sung Lee. He actually worked with many other researchers who are running the BioFoundry in the world, and he tried to standardize and integrate the BioFoundry workflow. Here is one example. When you make a small DNA construct, there are so many operations. So he defined level 0, level 1, level 2, level 3. He defined the operation at the different level. Then he tried to standardize all this protocol so that all the BioFoundry in the world can use this standardized protocol for the construction of the molecule or for the operation of the BioFoundry. And second thing is, probably many of you do this DNA assembly or the gene expression cassette construction in the lab. We are doing this by hand traditionally. And then, in order to make a transition from the hand-based cloning to this machine-based cloning, we need to optimize this assembly procedure. Here is one example. Even though it looks very similar, by locating the selection marker location, we found that this optimized integration method provides much better integration efficiency when we are using the machine. So definitely this human hand-based protocol need to be developed for the machine-based cloning. So this is another example. In order to overexpress multiple enzymes with the multiple promoters. Here is one example. This is a beta-carotene producing pathway with the 3 genes. In order to make this beta-carotene, we need to introduce 3 genes. But then we have this 5 to 6 promoters. So that means if we have all this permutation, you need to be constructing over 100 strains. If we do this by hand, maybe my grad student have to spend a couple of years. But now, using this biofoundry, we can construct all of this construct within a couple of weeks or even a couple of days. So here is one example. They constructed over 120 strain within a couple of weeks, and then they can monitor the production level, and they can figure it out which promoter is the optimum to make their target product. And this is another example. So, in case of the pathway optimization, we have this 100 or 1,000 library size. But if we do enzyme engineering, the size of library goes to 10 to 5 or 10 to 6. So we have even bigger, we have to deal with even bigger size of the library. But here, what they did was they were using AI-based approach to reduce the space of the enzyme sequence space, and then they build small subset of those enzymes, and then they measure the activity, and then they use those data for the next generation of the library. So by repeating this iterative cycle, they can find the optimal enzymes. Here is one example, is this ISPS, which is the key enzyme for making isoprene in like a plant and microorganism. This enzyme is notoriously have a low enzyme activity, but by doing this automated enzyme engineering procedure, they were able to find engineered enzyme has more than 5-fold increase. So by doing so, they can construct a strain which make better and more isoprene from the sugar. And also another important thing is that they can reduce the time. So the one key aspect of this biofoundry-based engineering is we can reduce the time of the development. So this is the achievement from the K-Biofoundry. I'm going to also give you an idea about this Agile Biofoundry, which is a US DOE-funded biofoundry. So this biofoundry has a consortium of 7 US DOE national labs. So as you know, there are many national labs. They wanted to have a standardized biofoundry or common biofoundry so that this biofoundry will serve the applied research in these 7 US national labs. And then this is the history. It was started in 2016, and they had continuous investment and reorganization, and then even transition. So still, it is quite working well, and then future funding is expected to continue this. I'm going to give you an idea of what they did the last couple of years, the last 10 years. So these are the recent achievements by the Agile Biofoundry. The first one is this genome scale tools. So I think this is one of the key contributions of biofoundry. Instead of working with a single enzyme, single gene deletion, now we can create genome-wide knockout library, genome-wide overexpression library, so that we try to characterize genomic library instead of just single cell. Here's one example. They did transposon mutagenesis with the barcode. So they created 4,000 or 5,000 strain has a knockout, and then they culture them under specific condition, and they can figure it out which gene knockout has advantage, which gene knockout has a disadvantage, so that they can study the function of the gene at the genome scale. And then this robot-assisted enzyme discovery, as I said, is quite important because the protein space, if you have like target enzyme and target protein, then each amino acid will have a 20 amino acid possibility. So if we want to make a 2 kind of site-directed mutagenesis, still 20 multiplied 20, you need to make a 400 kind of mutated protein sequence to figure it out their function. So the human hand cloning is quite difficult. So using this robot-assisted cloning, so you can create not 10 to 6 library, but still you can create much bigger the mutant library, so that you can use those library for the new enzyme discovery and engineering. And also another interesting thing we need to think about with the biofoundry is this host strain. So far, I was talking about enzyme and pathway, but this enzyme and pathway need to be evaluated on the different host. If we express this in the E. coli strain or yeast strain, mammalian host, depending on the host strain, the enzyme and pathway function could be changing. So that's why instead of just using one microbial host, we need to be considering many types of microbial hosts. So what they did was this tier system, tier 1, tier 2, tier 3, depending on their status of development, they define the tiers so that, in case of E. coli or Saccharomyces cerevisiae, this is a model system, could be the tier 1. But if we have a new microbial strain, could be tier 3. So by defining this tier, we know what should be done in order to make this tier 3 strain to the tier 2 strain to the tier 1. So this is also very interesting framework developed by Agile Biofoundry. And also this Biofoundry can be a tool for the machine learning. Because as you know, AI, everybody are talking about AI for the science, but the key aspect of for making AI successful is the availability of high quality data. Currently, all the biological data are collected from different labs. So that's why it's quite difficult to integrate those data. But if we have this kind of central machine generating the data, we can use those data for the machine learning. Here is one example. If we want to optimize a medium, if I use the data from A lab, which is published 10 years ago, and the data from B lab, which is published 3 years ago in a different country, unfortunately, we cannot combine these two data because they were done in the different lab, different condition. But if we are using one machine, and then we optimize a medium of condition, for the cell growth or for the production of our target product, we can do it much efficient way because we obtain high quality data which can be fed for the next cycle of the design. So this is a very useful area of using Biofoundry. And again, as I show you now, it's not about just enzyme and pathway construction. We can do strain construction optimization as well. So I'm going to talk about a little bit more in other slide. So another important kind of function of Biofoundry is a place where many people can meet and discuss. Here is one example. Agile Biofoundry is a kind of government Biofoundry service to many company, many university. So this is a location or venue where these people can discuss so that we can kind of help each other. So as you see, this Agile Biofoundry has many, many partners, including K Biofoundry and then I Biofoundry, where I'm working. So I'm going to talk about iBiofoundry. We are building, establishing for a decade. So as you see here, iBioFab, which was originally established in 2014, about 11 years ago. And then we had the continuous funding from the DOE and NIH and DOE, so we are able to renovate our biofoundry. And then recently, 2024, we got another major funding from the NSF, so we are able to completely rebuild the biofoundry. So these are the example of biofoundry work we did. The first one is bioautomata. So this is a paper published in 2019, but we already envisioned that self-driving laboratory can be implemented using this biofoundry concept. So here we kind of picked a target, which is lycopene production. And then we target this gene and promoter, and then using this biofoundry, we are able to run this DBTS cycle kind of as a closed loop, so that we can produce lycopene at a high amount without doing all the experiment. As you see here, we only did 1% of their kind of construction. When we talk about the pathway space, you have 3 enzyme, you have let's say 10 to 15 promoter, then you need to construct maybe billions of the construct. But instead of doing those millions of construct, we make a small set kind of representing small space first, and then use those data to guide the second stage strain construction. So by repeating this work, by only kind of searching the 1% of the space, we were able to obtain the optimum strain for producing lycopene. And then another important thing we are talking when we are building this biofoundry was the service. Many people think biofoundry could be very expensive toy of selected elite scientists. However, this biofoundry should be available to general public. So we build this plasmid maker. So this is kind of website-based plasmid construction, so the student can log in and they just pick like a gene and promoter, and they order this kind of gene construct through the website. Then they can get the delivery of the plasmid. We already have gene delivery or gene synthesis, but in this case, if you are synthesizing 100 construct with the same gene with 100 promoter, then you don't have to synthesize all 100 differently. So in this case, using this biofoundry, we can recycle those 100 promoter and the one of your gene, so we can easily construct 100 constructs. So this is kind of community resource. If you have a big research group, instead of having individual construct in each lab, we can have those as a community resource. Using this biofoundry, we can serve community much, much better. And another thing is the biofoundry does not provide everything. We need this kind of AI tools. Here is one example. This protein structure prediction has been a big problem. So as you know, this AlphaFold happening. So now, this Nobel Prize winning computer software is very good at predicting the 3-dimensional structure of the protein, but still the prediction of function is quite difficult. So what we did was Professor Humin Zhao in our university, he created this very interesting AI algorithm, CLEAN. So using this CLEAN, now we can predict the function of the protein. So, this is already on the service. So if you type this URL, you can access. If you enter your protein sequence here, we can predict EC numbers. As you know, all the enzyme has EC number. We can predict this EC number quite accurately. So this is another AI tool so that we reduce the search space or library space in order to make our biofoundry-based engineering to be more effective. So one of the kind of the transition we made this year is this research-oriented biofoundry. So until 2024, the biofoundry we have in Illinois was mostly for the scientists in Illinois. But we wanted to open this biofoundry to everybody. So now we are changing to service-oriented biofoundry because now we obtained substantial expertise how to run the biofoundry. So we want to use this expertise to serve like broader community in biotech. So we got the funding from NSF. So this is the kind of our plan, the phase 1. which is last and this year, what we are doing is we build a new biofoundry, and then we, as I said, this plasmid maker and CRISPR copies or automated gene knockout or automated mutagenesis workflow. We already developed this from our research biofoundry. Now we are porting those into our service biofoundry, so our customer or users can use this so that they can do their research. And then eventually, we would like to provide all of these two as a kind of one-stop shop for strain engineering, so that become a very useful kind of resource for all the biotechnology research scientists. So again, our biofoundry is a nexus point for the collaboration. So we are working with the other biofoundries such as Agile Biofoundry and K Biofoundry, and also we are working with the company and users, and also many other the stakeholders like citizen scientists, and we also offer training and education of non-scientists as well. So with this kind of biofoundry, this project, use this biofoundry framework for sustainable biomanufacturing. So I'm going to explain a little bit more about this project here. So we wanted to make this terpenoid production system. And so if I give you a little bit more detail, so this is the pathway we are talking about. So here, as you see, isopren or isopreno, which is shown in the center or at the end, these are the two molecules which can be produced by the microbial strain. But all the microbial strain making this product in the nature accumulate IPP, isopentanylate pyrophosphate, which is shown in the bottom, which is very toxic to the cell. And also this pathway requires use of ATPs, as you see in the red. So this is a very energy intensive pathway and also involving toxic intermediate. So in order to overcome this kind of problem, our project, think about this novel idea, which is to bypass natural pathway and create artificial enzyme which produce isopreno more efficient way. So one idea is instead of using existing enzyme, we discover and invent a new enzyme which has a different reaction mechanism. Here we use this kinase plus decarboxylase enzyme mechanism so that we can do much better biocatalysis as compared to the enzyme existing in the nature. And in addition to this strain engineering, as methane as a substrate has very low solubility in the water. So we also need to optimize the culture condition or optimize the bioprocess condition to increase the solubility or increase assimilation of methane. So we are working for that problem. And this is something we already achieved. So now we have this automated gene library construction. And also we have all this kind of semi-automation kind of system. And also we obtained more instrument to make a better biofoundry. And so these are the three kind of area of global collaboration with the ABF. So now they're developing the system and then obtain the fund for cell-free system establishment. And with the Illinois, with the iBioFab, we are working to use the enzyme discovered from K Biofoundry, and then we use that in the heterotrophic microorganism instead of methane using microorganism. And also the collaboration with Canada, they are developing bioprocess optimization. Also the Canadian PI is also identifying a new enzyme for make various carotenoid product. And one of the culmination is, with this kind of ongoing collaboration, we are able to have this biofoundry session in the US-Korea Synthetic Biology Conference this year in Champaign. As you see here, we had this session, and then we are talking about how to run the biofoundry together so that we share the resource. We do not repeat the mistake so that we can effectively run the biofoundry together. And this is our expected the outcome of this project. So we build this network with the US and Korea biofoundries, and then we develop the protocol workflow so that we can share those protocol. And also, particularly we want to develop the enzyme and pathway for the efficient production of useful chemical using methane as a substrate. So I'd like to thank all the participants here, especially Dr. Seung-gu Lee, who is a leader of this whole project, and also Professor Byung-Ha Ho from KAIST, and also the Agile Biofoundry, Nathan Hilson, iBiofoundry, Hugh-Min Zhao, and Stefan Lane, and also Sun-Won Kim from Gyeongsang National University, and also Tae-Gyun Lo from University of Calgary. And also I'd like to thank funding agency, and also I'd like to thank the US funding agency supporting ABF and iBiofoundry. I think this is the end of my slide. So thank you for your attention, and happy to answer your question. Thank you.
Thank you very much, Professor Chin, for your insightful presentation. We look forward to deepening global collaboration among the biofoundries and to seeing these efforts expand into wider applications in synthetic biology, food, and pharmaceutical innovation. We will now open for discussion. Any questions from floor and online?
Thank you, Professor, for the inspirational talk regarding this AI-based engineering. So I am questioned that, like you mentioned, the importance of the AI-based engineering, and I want to know more about how Biofoundry can facilitate based on this AI-based engineering part, like how we can use Biofoundry to facilitate this AI-based engineering work.
So this is a very important question. We're debating how to use Biofoundry for facilitating this AI-based biotechnology. And as I said before, I think there are two kind of utility of Biofoundry. The first one is as a source of data. And as I explained already during my presentation, traditional data generation in biology was based on the lab notebook. I have this 20 lab notebook from my student. I remember some of them, but I cannot remember all of them. And some of them are connected, but human recognition cannot recognize. But same thing, but if we use Biofoundry, all this information will be digitized because this is machine-generated data, so that we can archive biological data much better than human-based experiment. So we can obtain high-quality data, and also all the experiment was done at the same condition or similar condition by one machine. So we have a high-quality data generation possibility using the Biofoundry. That's one kind of utility. And second is for the implementation. Once you figured it out something, because always this AI-based approach require iteration. Probably you know that even ChatGPT, version 1, version 2, version 3, by running this cycle, the AI program getting smarter and smarter. Same thing, once we obtain the initial data, we analyze them, then we make a subset of the data, we feed those into Biofoundry, and we get the data again. By doing so, we can improve this whole AI process so that we can have a better and more accurate prediction. So I think those are the two utility of Biofoundry in the time of AI-based biotech research.
Yeah, pass the microphone.
Thank you for your presentation. My question is that I'm an industrial worker. So Korea is as famous for the regulation system as for the K-pop demon hunters. So my question is, how do you think about that? The Biofoundry based on strain engineering can be used in the non-GMO industry area?
Oh, yeah, that's also a very good question because, still in order to apply, genetically engineered microorganism for food and certain area, maybe even in medical, sometimes we have a kind of a bottleneck. So, biofoundry is also can be used for like laboratory evolution. We can do evolution experiment, which is quite easy to implement. So instead of targeting a certain enzyme and gene, we can use laboratory evolution using the biofoundry so that we can screen many strains under evolution condition so that that kind of adaptive evolution experiment can be done by biofoundry. And second is actually one of the area, I have a colleague who run the yeast company. So they collected thousands of yeast strain from nuruk, from the doenjang, from the pine tree, and they collected many yeast strain, a couple of thousand, and they were testing those 5,000 yeast strain under certain culture condition to figure it out which strain is making most to heme or most to certain metabolite. So definitely this high throughput screening of naturally existing strain, biofoundry can be contributing, which may be a very good use of biofoundry for industry or fermentation industry or food industry now.
If I may, I would like to ask one last question myself. I recently attended the IGM Jamboree held in Paris last week. The global synthetic biology competition that had begun from 2005. It has given rise to many pioneering startups such as Ginkgo Bioworks, OpenTron. This year, around 5,000 participants from 400 teams took part in there. Professor Jin, how do you view the stimulating role and impact of those IGM in advancing synthetic biology?
Oh, yeah. So we talk about the importance of data in AI era, right? But I think the most important thing more than data is the next generation workforce. If we do not have a next generation workforce, highly motivated workforce, we cannot sustain this R&D. So in this area of microbial engineering or synthetic biology, I know this iGEM, which is the biggest competition, like robot soccer, like 20 years ago when I was like college student. I remember that every university tried to compete for this robot soccer game. I think the robot soccer game actually had a big outcome. Due to that kind of interesting event, many students want to major ECE or computer engineering, so which is actually the kind of foundation of IT industry or semiconductor industry of this country. So I think it will work same way. So this if iGEM is more activated in this country, which will kind of increase the visibility of biotech research, and also it will help recruit highly talented students into this area, so that 10 to 20 or 30 years later, our country or the Korea would be another make another innovation in this biotech area, like the semiconductor industry right now, what we are doing. So yeah, definitely this iGEM is quite important, and I'm kind of so sorry to hear that only one team was attending this year. So hopefully this kind of activity can be activated or can be supported better, so that more and more students can attend this kind of activity, which is also another thing we need to think about. Probably one of the big debate these days is like this medical doctor versus scientist engineer. Like many high school students, talented or smart high school students want to be a doctor, medical doctor, due to their stable life and income. But I think that's one of our kind of mistake. We should show more vision and more vision or the futuristic kind of event like iGEM, so that we can recruit more talented students into this field. I think that's something we need to do.
Thank you. I agree. As we are approaching to the end of this session, let's conclude here. Once again, please join me to give a warm round of applause to thank Professor Jin Young-soo for his excellent presentation and discussion. Thank you.
Thanks once again, and thanks to our moderator and speaker, and please give another big round of applause. Now this concludes the session 3 of the symposium, and we will now have a lunch break. Before you leave, let me briefly guide you to the lunch venue. There are same restaurants as yesterday, and you can enjoy lunch using the lunch coupons that have been provided at the registration center. And dining is available at restaurants located on this floor on B2 of COEX Mago Convention Center, and it will be on the screen. And the restaurants are the same as yesterday. And please enjoy your meal and take a pleasant break. And our afternoon session will begin at 2 o'clock. Thank you very much and see you soon. And for green name cards for VIPs, can I ask our guests to move to the Sanghae-ru, which is right in front of our conference hall. And for those who have green name cards, we kindly ask all participants with green cards to move to Sanghae-ru right away. Thank you very much. Please feel free to use the receiver on the desk or at the back of the conference hall, channel 1 for Korean and channel 2 for English. And in the Square Volume lobby, you can enjoy various programs, including regional achievement exhibitions, a vision promotion, video, and a photo zone. We encourage everyone to take interest and join in. And for lastly, please check once again if you set your mobile phone to a silent mode. And please note that today's presentation materials are provided with e-book format via QR code. You can scan the QR code to download the presentation materials. 네, 마지막으로 저 명찰에 대해서 다시
And once again about the name tag. You cannot use the same number as yesterday for the lucky draw event that will take place in the end of today's event. So please make sure that you proceed to the registration desk to issue a new number. And also with regards to the satisfaction survey, there will be QR codes in this room and outside the hall. Please participate in the satisfaction survey, and at 2, session 4 will shortly begin.
Welcome back, everyone.
네, 여러분. 저희가 이제 세션 4를 시작하도록 하겠습니다. 네 번째 세션은 EU 권역이에요.
Digital Health, Advanced New Drug and Gene Therapy. And each theme will include presentations followed by a short Q&A session. And this session will be led by Professor Hyunwook Kim from KAIST. And please welcome our moderator with a big round of applause.
Okay, good afternoon. Thank you all for being here in the room. We'd like to start the afternoon session. This is the collaboration with the EU teams. The first speaker in the afternoon is Professor Kang Hak-soo from Konkuk University, who's standing right in front over there. So the name of our research center is Korea EU Synthetic Biology Research Center. So we are collaborating with the Technical University of Denmark in Denmark. We are focusing on microbial synthetic biology for the enhanced production of natural products with the medicinal potential. I am the PI of the research center, and Professor Kang Hak-soo, whom I invited for this symposium, he's the co-PI for this project, and he is one of the best, I should say, the best expert in the field of natural product synthetic biology. He will be talking about various molecular tools, synthetic biology tools, that will allow the enhanced production of our target natural products. Without further ado, please join me welcoming Professor Kang Hak-soo.
It's my great pleasure to present our research in this symposium. So today I'm going to talk about our synthetic biology approach for the discovery and the production of natural products. So natural products are a very important source of drugs. So few examples are listed here. And the first example is daptomycin, which is used as antibiotic. and very active against the multi-drug resistant bacteria. And other example is FK506, which is immunosuppressant, and it's used to suppress the immune system. And another example is caliciamycin, which is a very strong anti-cancer agent. And in the clinic, it's used as the antibody drug conjugate. And the problem with the natural products is very low production of the natural product, very low production by the native strain. So in traditional way, all of these natural products has been produced by chemical synthesis. But as you can see in this slide, the natural product has more complex structure compared to the synthetic drug. So the total synthesis is not very efficient. So for example, the vancomycin requires 30 different chemical steps, and the overall yield is less than 1%. And also the rapamycin requires 27 steps, and overall yield is very low. So this problem has been very challenging in the drug discovery process using natural products. After the discovery of active compounds, then the most important problem is the supply of active compounds in the clinical trial, and also the delibertization becomes very difficult. And all of these problems have been addressed using organic chemistry, but our team works on synthetic biology to address these important problems. So today I'm going to talk about two different stories. And the first story is about the development of industrial strain for daptomycin production. And daptomycin is a blockbuster antibiotic, and it's produced by Streptomyces rogeosporus, and the global sale is around $1 billion, and most of the sale occurs in United States. But the production titer by wild type strain is very low. It's around 20 to 40 milligram per liter in flask culture, and 100 to 300 milligram per liter in batch fermentation. So because of this low titer, the production cost is quite high compared to the vancomycin. And this is how daptomycin is produced in commercial scale. The daptomycin has a lipid chain, which is a straight chain decanoic acid, but this fatty acid is not able to be made by native strain. So we have to feed this decanoic acid into the fermentation media. Then this strain produces the mixture of daptomycin together with the natural lipopeptides. So our first goal to develop the industrial strain is to improve the overall production titer of the daptomycin, of the lipopeptide. And this daptomycin is produced by the three different biosynthetic steps. The first step is lipidation, which incorporates fatty acid into the peptide chain. And the next step is peptide elongation, which produces the linear chain peptide, and it's finally cyclized into the cyclic lipopeptide. And also the precursor biosynthetic genes are present. And all of these biosynthetic genes are located together in microbial genome, forming biosynthetic gene cluster. So first we performed RNA-seq analysis to see these biosynthetic genes are transcribed well, but the RNA-seq analysis shows that the transcription level is very low. So we used this our promoter engineering platform to refactor the biosynthetic gene cluster. So first we developed the synthetic promoter library, and then then classified them into the strong, medium, and weak promoter using our promoter assay platform. And then we refactor the biosynthetic gene cluster using our promoter library. That we constructed a series of different combinations of promoter and compared the production title. And the construct showed the high transcription level, and finally it showed the higher production of daptomycin. The wild type title was around 26 milligram per liter, but after promoter engineering, then we achieved the gram per liter scale production of lipopeptides. So after promoter engineering, this is the lipopeptide profile that we have in the broth. Then it produced daptomycin together with 4 major natural lipopeptides. And except for daptomycin, all other lipopeptides are very toxic. So because of the presence of toxic lipopeptides in the broth, the purification process costs a lot of money. So overall production cost is high because of the complex purification step. So if we can develop the strain that produce only daptomycin without producing toxic lipopeptide, we can significantly reduce the overall production cost by simplifying the purification step. So our next goal was to eliminate all the toxic lipopeptide and only produce daptomycin. So to achieve this goal, we have to engineer the overall fatty acid metabolism in the producer strain. And the fatty acid metabolism is composed of primary and secondary metabolisms. And the primary metabolism produces the pool of fatty acid in the cell, and the secondary metabolism picks up the fatty acid depending on the substrate selectivity and load into the biosynthetic enzyme to produce the final compound. And the first step to engineer the lipidation process is to increase the selectivity of fatty acid. So because the mixture of lipopeptide produced in the strain is because of the broad substrate specificity of enzymes. So if we can engineer the enzyme to increase substrate specificity, then we can eliminate the production of the lipopeptides. And in daptomycin biosynthesis, and this enzyme, the fatty acid AMP ligase is responsible for selection of fatty acid for the lipopeptide production. So we searched the database to find the functional homologue with the higher selectivity. Then we searched almost the whole database that produce the decanoic acid, and then found these 4 functional homologues. And then replaced this enzyme with the functional homologues and then compared the lipopeptide profile. And the one enzyme, HMQF and the CYLA, it shows the higher selectivity against the decanoic acid. Then after replacing the native enzyme with HMQF or CYLA, they only produce daptomycin without producing the toxic lipopeptides. So, but when we engineer this enzyme, then we faced one problem because the fatty acyl AMP ligase interacts with the acyl carrier protein by protein-protein interaction to find for the biosynthesis of lipopeptide. But if we replace this enzyme, native enzyme, with a foreign functional homologue, then we lose this interaction. So we thought about how to overcome this problem. Then we tried the fusion of these two enzymes to one protein. Then this enzyme that are fused between fatty acyl AMP ligase and the acyl carrier protein, so significantly improved the production of daptomycin. So we obtained almost 4 times higher the production with the fusion proteins. So by engineering the fatty acyl AMP ligase gene, then we were able to produce only daptomycin without producing the toxic lipopeptides. So the next step is to engineer the primary metabolites. So even though we have the high production and the high purity production of daptomycin, the one final problem is that we still have to feed the decanoic acid into the fermentation broth because this strain doesn't naturally produce decanoic acid. So next step was to engineer the primary metabolites so we can achieve the biosynthesis of decanoic acid in the producer strain. So because the producer strain only produces the branched chain fatty acid, so to produce the straight chain decanoic acid, so we have to modify this fatty acid metabolism. So our first the step was to feed the long chain straight chain fatty acid. Then it can enter beta oxidation pathway and then produce decanoic acid. Then when we feed the long straight chain fatty acid, then it successfully produces daptomycin without feeding decanoic acid. So which means that if we can produce long chain fatty acid in the strain, then we can produce daptomycin without feeding the decanoic acid. So we looked into the bacteria. Then we found these 2 bacteria, Mycobacterium and the Corynebacterium. They have this type 1 fatty acid synthase, which is the multimodular, the mega synthesis. And then they are very good at producing long straight chain fatty acid. So we chemically synthesized this gene. It's about 10 kilo base pair. And then we combined with the different strengths of promoter and constructed the pathway, type 1 fatty acid pathway. Then we inserted this type 1 fatty acid system into the producer strain, and it successfully produced daptomycin without feeding decanoic acid. And the production level depends on the promoter strength of type 1 fatty acid synthase gene. So for the first parts, so by 2-step engineering, so we were able to produce daptomycin with a high titer and a high purity and naturally without feeding decanoic acid. So now the overall production titer of daptomycin in the fermenter is about 3.5 grams per liter. So now we are collaborating with the biotech company to commercialize this strain. We are going from 5 liter lab scale to 300,000 liter ferment, the pilot scale by the step by step. And the second part is about how we discover new compound and deliver ties into the various structures. So several years ago, one paper was published, and this paper was about if we inactivate the gene cluster of common antibiotics, then we can reveal the hidden antibiotics that are silent or cryptic in the microbial genome. So we want to try this concept. So we took this daptomycin producer strain and deleted this daptomycin, the biosynthetic gene cluster. And the deletion of this gene cluster improved the production of 3 different types of metabolites. Then we try to connect this biosynthetic gene cluster into the metabolites, then using LC-mass analysis. And the metabolite class B was connected to the cholismycin, and the metabolite class C was connected to allelomycin. But the class A was not able to be connected to any type of biosynthetic gene cluster. So we solved the structure using the NMR. Then we found that this metabolite is a new compound composed of nucleotide moiety and an amino acid. And this structure was quite novel. And the only similar compound is equiguanine, which was published in 1991. And also it's the most potent inhibitor of PI3 kinase. So because of this compound, there's this CDG moiety, the carboxydiazoguanine moiety, and this metabolite is known to be synthesized by 3 different biosynthetic enzymes, including 4E, the QUED, and QUE, and all of the natural product that has this moiety has these 3 genes in the biosynthetic gene cluster. So using these 3 genes, we identify the biosynthetic gene cluster of our new compound, and which is we named rhodamycin biosynthetic gene cluster. To confirm that this biosynthetic gene cluster produced these metabolites, so we cloned this gene cluster by the direct cloning in yeast, and then they expressed in the heterologous host, the Streptomyces albus. Then it produced the same compound and also produced the more complex compound called 2B and 3B. And this compound has the nucleotide CDG moiety and 2 separate amino acids. So we generated a series of the deletion mutants to figure out the biosynthetic pathway of this compound. And then this compound has 2 different amide groups. And one is the amide group between 2 different amino acids, so beta hydroxyl asparagine and desaturated lysine. And then the other amide bond is between desaturated lysine and the CDG, the nucleotide core. And these 2 amide bonds are catalyzed by 2 different enzymes. And the first enzyme is the ATP grasp enzyme, which is responsible for amide bond formation between asparagine and lysine, and the amide bond synthetase, which is responsible for the formation of amide bond between lysine and the CDG core. And the ATP grasp enzyme was very common, and it's found many times in natural product biosynthesis. But the second enzyme, amide bond synthetase, which is very rare, and it's very rarely found in natural product biosynthesis. And so we wanted to study more about this enzyme, amide bond synthetase, and the closest functional homologue was the TOEM enzyme, which is responsible for nitric formation in toyocamycin the biosynthesis. And rhodamycin, which is our compound, has amide bond, but toyocamycin has a nitrile moiety. So we want to study what's the relationship between these 2 enzymes. And in case of toyocamycin, this nitrile group is formed by 2 different steps. And in the first step, this TOEM enzyme uses the CDG as a carboxyl substrate and the ammonia as amine substrate to form this amide intermediate, and which is further oxidized into nitrogen moiety. So our hypothesis was that these two enzymes had the same catalytic reaction, but with a different substrate profile. The ROEL used the lysine as an amine substrate, but TOEM used ammonia as an amine substrate. So we verified this hypothesis using in vitro enzyme reaction. We tested 4 different substrates for TOEM or ROEL, and TOEM shows very high substrate specificity to the ammonia, but the ROEL showed high substrate specificity to L-lysine or N-acetyl lysine, so which confirms our hypothesis. So TOEM and ROEL are both amide bond synthetase, but they have very different substrate from the specificity. Then we used this alpha fold 3, the structure, to find the active site. Then we combined alpha fold 3 with the Autodoc to analyze the active site and then confirmed the possible candidate of catalytic amino acid residues. Then we generated a series of the single mutant, and then by analyzing the production of metabolite, we were able to identify the key catalytic residues for this enzyme. Then because of this enzyme is quite novel, so we wanted to find more of these enzymes by searching for the microbial genomes. So we used 2 different tools, the EFI tool and the BLAST-P, with 30% identity as a cutoff. And then we found the 43 functional homologs using EFI tool and the 680 homologs using BLAST-P. We narrowed down to the enzymes that are only present in the Streptomyces, and then by phylogenetic analysis, we will divide them into 4 different clades. And then we expressed this functional homologue with the CDG cassette made of 3 enzymes. So that produced CDG carboxyl substrate, and then by the last functional homologue enzyme, they form this new amide bonds. Then by solving the structure of metabolites, they were able to diversify the structures. The clade 1 homologue used ammonia as amine substrate, and the clade 2 homologue used lysine as a substrate, and clade 3 used the 1 carbon less than the lysine, and then the clade 4, which used D-alanine dimer as an amine substrate. So in summary, we found this new enzyme. We call it D-azapurin amide bond synthetase, and this enzyme family uses CDG as a sole carboxyl substrate and diverse amine substrate to produce diverse structures of the D-azapurin natural products. So this is the summary of the second part. Then by deletion of daptomycin biosynthetic gene cluster, so we produced a new compound, and then in the biosynthesis of this compound is catalyzed by novel enzyme. We call it DABS enzyme, and by searching for database, then we were able to successfully diversify the structure by gene mining. So finally, I briefly introduced about our center. Then the PI of our center is the Hyunwoo Kim from KAIST, and our center is based on the long-time collaboration between the KAIST and the DTU, between the Professor Hyunwook Kim and the Professor Tilman Weber. And we established the biofoundry in KAIST, and then also we are exchanging students. And one DTU student came to Korea, and 2 Korean students went to the DTU to conduct research collaboration. And also we published the collaborative paper together with the other paper. And also we hosted 5 different seminars and 1 international symposium. And the collaborative research is still going on, so probably next time we can talk about our collaborative research. Then, thank you for your attention.
Thank you for a great talk and also a kind introduction of the research center. We can take a couple of questions from the audience, if you may have any. I'm aware that you come from different backgrounds, but nonetheless.
Thank you for a great talk. And I have a question about the prioritizing the biosynthetic gene cluster product. Since there are abundant amount of gene cluster hidden in the streptomyces or other microorganisms, I want to know if you have any criteria for prioritizing some BGC before you get into digging into the BGC product.
I think that's a great question. And as far as I know, the many groups had different approach to prioritize the biosynthetic gene cluster. But in our lab, we prioritize based on the presence of novel enzymes. So we can do this kind of study because the novel enzymes are very important. So you can use this enzyme to produce compound and also to engineer this enzyme by directed evolution to diversify the structure. So the presence of novel enzyme is our first priority when we prioritizing biosynthetic gene cluster.
Thank you for the answer.
I have a question.
Yeah.
So we want to find out a new molecule. Then we could think of 2 different approaches. One is to genetically engineer the biosynthetic gene cluster for a gene cluster that we already know. Another one is expanding the size of microbial library. Which approach would be more efficient and effective from your point of view?
So I think it also depends on depends on the group. Some group prefers the high throughput approach, and some group prefers the very specific approach. But in our group, I think every biosynthetic gene cluster has different problem. So we only focus on, I think you can use the bioinformatic tool to prioritize biosynthetic gene cluster, but the production of metabolite shouldn't be generalized. Any biosynthetic gene cluster has a different problem, so we have to focus on the biosynthetic gene cluster, not the scale. That's my opinion, but some people have a different opinion.
Appreciate it. Before I close the talk, just in case. Nope. So then let's thank Professor Kang once again, and thank you all.
Thanks once again. It was a session under the theme Synthetic Biology, and please give another big round of applause to our moderator and speaker. Now it's time to move on to the next session theme, and it will be under the theme Digital Health, and we have prepared one presentation for digital health, and I'd like to invite our moderator to the stage, and it will be led by Dr. Jong-chul Ra from Korea Brain Research Institute, and please welcome him with a big round of applause.
Thank you. I'm Jong Chul Ra, a group leader at Korea Brain Research Institute, and I'm currently participating in a collaborative project with the Netherlands Institute of Neuroscience. It's called Korea-Netherlands Mind Mirage. And in this project, we aim to understand the underlying principles and mechanisms of hallucination. And our collaboration with the Institute of Neuroscience began with the human brain tissue collected from the Netherlands Brain Bank. We're trying to focusing on the molecular analysis and histological characterization of these brains. And recently our partnership began to expanding into neurophysiology, where the scientists in NIN brought a great expertise. And also our partnership has extended to neuroimaging. And in this field, our speaker today, Dr. Serge Dumoulin, plays a central role there. And I'm very pleased to introduce Dr. Serge Dumoulin, a group leader of Netherlands Institute of Neuroscience, and director of Spinoza Institute of Neuroimaging. He did his PhD in McGill University, Canada, and then he did a brief postdoctoral training in McGill and another training in Stanford University, and then came back to Netherlands in 2008, became a professor in Utrecht University in Netherlands. And from 2016, he's serving as a director of Spinoza Center for Neuroimaging, and he's going to talk about psychedelic in context, vision of altered minds. So without further ado, please welcome Dr. Dumoulin.
Thank you very much, and thank you very much for organizing and inviting me. And it's a pleasure to be here, and it's a pleasure to collaborate with the Korean Brain Research Institute. Starting off with a very basic question, why would we study the human brain? And I think the brain really determines all of our actions, our perception of the world, and really determines who we are. So the study of the brain is not only a study of a specific organ, but it's also the study of ourselves and how we function in this world. Brain disorders have a profound impact on our society, and with about 8 billion euros per year in Europe alone. And I think it's important to note that for many of these diseases, there's no cure. About 1 to 5 people will develop some kind of dementia when we grow older, and we will have to learn to live with this. As our youth develops, a lot of them develop psychiatric disorders such as schizophrenia, autism, bipolar disorder, depression, and there's also, they're going to have to learn to live with this and cope, but there's no cure. So this is the focus point of the Netherlands Institute for Neuroscience, to study our mental functions. And this is something we share with the Korea Brain Research Institute. And in particular, in the Netherlands Institute for Neuroscience, we really want to push the research line towards psychiatry. And with the idea, what psychiatry are also mental illnesses and disorders of brain function. Now, I will talk about my own research line in this and how I've collaborated with this, which is using psychedelics and in human subjects. And so what was the point of studying psychedelics? Well, a lot of mental illnesses are associated with hallucinations, something that we also just referred to. So hallucinations are very common in a lot of brain disorders, and especially psychiatric disorders such as schizophrenia, bipolar disorder, major depression disorder, but also in more neurological disorders such as dementia. And the reason to study psychedelics is that psychedelics actually induce hallucinations and therefore perform a model of how hallucinations may occur. And so really then the focus of the research is, okay, what is the mechanism by which the brain produces hallucinations? And the focus of my research group is then psychedelics like psilocybin help us to model and understand hallucinations, and can we study the mechanisms of hallucinations using psychedelics? A little bit of psychedelics that we use, it's psilocybin. That's the main ingredient of magic mushrooms. And the hallucinations that people report are basically the world starts moving around, the object starts breathing, walls start melting, you see all kinds of surreal geometric patterns. And this is the kind of thing, so, okay, so it induces these types of hallucinations, how does it produce these? The other thing I should mention about psilocybin, it also is actually put forward, sort of paradoxically, as a treatment. And so people are now going through clinical trials and saying, okay, how can we use psychedelics to actually improve brain function, especially in, for example, depression, anxiety, or post-traumatic stress syndrome. So really, sort of the goal is really to study hallucinations, a common feature of brain disorders, which is seen in many brain disorders, and psychedelics can help us to model and understand these hallucinations. My background is really in vision, so I really want to use the visual system as a model to understand psychedelics, and particularly psilocybin and how this operates. And vision, one important aspect of vision, and I think life in general, is that context shapes vision. And this is what you see here is a very common visual illusions. The two orange circles are actually the same size. So if you remove, if you ask people to judge which circle is bigger, people will often choose the one on the right. And if you remove the surrounds, then you will actually see they're actually the same size. So this is a visual illusion. And it actually shows us that visual perception is determined by the context or the surround. And so context affects perception. This is called the Ebbinghaus illusion. This is a well-known illusion. And context determines perception, but you could say, well, context shapes almost everything in life, right? So in my group, we use vision as a window into the brain function, and one of the important aspects of vision is that context shapes vision. So how does context shape neural responses? So this is one of my favorite sort of experiments. What you see here is a subject looking at the red dot on this visual stimulus. On the left, on the right, you see the right hemisphere, the brain of one particular participant who's lying in the fMRI scanner. And you can see the activity progressing as the stimulus also progresses. So you see this expanding wave on the brain, while you also see this expanding, the ring expanding on the visual screen. And this is, this experiment was performed right after the invention of fMRI, which is in 1992, the bold contrast. This was published by Engel in Nature in 1994. Built on by Serino in Science in 1995, and I'm still doing these kind of experiments. And one important aspect is really, if you look at the visual cortex, that neurons respond to a specific part of the visual field. And you can see that here. So this is the population receptive field. So you see when the stimulus go through the population receptive field, you get a response. So that means that if we look at a visual scene such as this is my kids, then we have the neurons populations process the visual image locally in these population receptive fields. And the population, in this case, refers to a population of neurons. So this means that every neuron has a receptive field, and this is just a net receptive field that we can measure with brain imaging tools. And we can measure them now reliably under half an hour in every participant. And in my group, we built models of what this population receptive field looks like. And this is very briefly explaining the model procedure. So we show a stimulus. We actually use bars instead of circles. We record some fMRI signal. So this is the signal of one point in the brain. And then we build a model of what the population receptive field looks like. And the typical one is a one-dimensional Gaussian. And if you have the model, you can actually predict when it would respond. So when there's overlap between the population receptive field and the stimulus, then you would expect a response. Now then you can actually predict what the response looks like. If that is not a good prediction, then you can say, okay, maybe I should change my population receptive field parameters to get the optimal fit. So this is a basic forward model fit, minimizing the residual sum of squares. And you can do this across all the brain, and the brain signals actually looks quite different, but the model captures all these different variations. So this is a good model of throughout the visual cortex, I should say. And you get basic parameters out. So X and Y give you visual field maps. So the visual field, the visual space is not processed once, but multiple times throughout the visual cortex, and we study what these areas are doing. You also get population receptive field size, so this is the area that they process, and you see that in later visual areas, the area gets systematically bigger, so this is a very systematic variation. So the next question is, okay, does context actually affect these population receptive fields? So population receptive fields is a really local processing of the visual scene that happens in about, the 25% in the back of your brain. So does context affect it? So the sort of the surrounding areas, as you saw in the illusion. Now there's a very influential algorithms proposed about how the brain processes context, and this is by divisive normalization. So basically, neuronal populations, or neurons in this sense, they get some input, especially in visual system, and they give some response. But the idea is that this response is modulated by the context in a divisive normalization. And this can be the context in visual space, but this can also be the context in neural space. So neurons are modulated by their neighbors, right? So it doesn't matter how much a neuron fires or is activated, it matters how it's activated relative to the rest. And so responses really in this model are normalized by context. And so we actually devised a population receptive field model in the same way, where we say, okay, the population response is actually made up by two populations and they're divided by each other. And in this case, I should mention Marco Akil, who is my PhD student, who actually did a lot of the work I'm presenting today. And so basically, this is the equation in a very sort of schematic diagram. So you have two Gaussians that are divided by each other, and they have two constants. And typically, the constants are added to prevent division by zero and to compensate for things. But these constants turn out to have very particular and important roles. So for example, if we vary the B parameter, you see that the Gaussian population receptive field shape actually changes to a Mexican hat shape. And really it is the B parameter that controls this Mexican hat shape. So you get decreases responses in the surrounding areas. Now the question is, this is all model-based, this is all sort of algorithmic-based. Does this actually happen in the brain? And so what you see here is again a little illustration of a time series or response in a particular part of the brain. And you can see this bar sweeping through the population receptive field, and you see these responses occurring that actually match. But you also see that before you hit the population receptive field, you actually get these negative or decreases in the response. And that are not fit by this model. So, and then we say, okay, if we fit it with the divisive normalization model, can we explain these decreases in the responses? So if we fit it, we refit with the divisive normalization model, and then you get the fit with the solid line on the right. And you can see that now we can actually capture these negative or these decreases in the neuroimaging response. And then you see that the PRF actually changes to a center surround configuration, this Mexican hat, where the surround is actually surrounding the population receptive field. And we've measured mainly with fMRI, but these PRF models, they actually bridge different neuroimaging modalities and different perception. So what you see here is the fMRI response. This actually, in this case, was a particular patient, so a patient with epilepsy who then was implanted with electrodes. And this was for pre-surgical planning. So they have these electrodes planted, and you can also measure these responses with these electrodes that are now directly placed on the brain. And on the right, you see the frequency versus time responses. And you can see, as the bar sweeps through the receptive field, you also see in a broad range of frequencies a response. And if you fit a PRF model, then you see that the electrophysiology responses actually match very closely to the fMRI, the bold responses. And except for the context is actually reflected in negative bold and in very specific frequencies in the frequency spectrum, and these are the alpha frequencies, they're called the alpha frequencies. And to many, this won't come as a surprise because the alpha frequencies are typically associated with suppression in the brain. Now, if you have electrodes on the brain, the other thing you can do is you can also stimulate. And if you stimulate these electrodes, people start seeing phosphine. So this is flashes of light. And here you can see where the ECOG electrodes were in the visual field, or the population receptive field, and where the subject actually drew where the phosphines were. And this is work by John Winnower. And then you can see that these flashes of light actually correspond to the location of the population receptive field. So these population receptive fields really bridge sort of different neuroimaging modalities, different brain imaging modalities, and towards perception. So what I really want to say in this little section is that the brain uses divisive normalization as a sort of contextual algorithm, or at least the algorithm actually captures the behavior of the neural populations. And these these kind of observations actually transcend different neuroimaging modalities. So the next question we ask is, okay, how is divisive normalization actually implemented in the brain? So what we had is an algorithm, but what is the biological implementation? And for this, we actually had a lot of combined two data sets. So we had a data set of about 180 participants who are doing these population receptive field mapping experiments. And from that, you can actually reconstruct a visual field representation. You can actually do this model fit of this divisive normalization. So here you can see how the activation constant actually varies across the brain, and similar for the normalization constant. So you fit this model independently for every point in the brain, and here you can see how the model parameters vary across the brain. Now we correlated this with published PET neurotransmitter atlases. So on the left, you see the fMRI data, a completely different data set, completely different participants, and on the right, you see now these PET neurotransmitter atlases. So you see the GABA distribution and different serotonin and neurotransmitter receptor distribution. And one thing you can already see, if you bring them in this common space, the neurotransmitters, even though they are not actually mapped for visual cortex, do sort of reflect some features of visual cortex. So now we basically put the two together and say, okay, do these model parameters that are actually independently fit correlate with these neurotransmitters of the other data set? Now if we bring them all in a common space. And you can see this really beautiful correlation. And on the y-axis, you see the different model parameters, and the two x-axis, the two x or z-axis, as you will, you actually see the different neurotransmitter components. So you see actually there's a very beautiful correlation between the model parameters on one hand and the neurotransmitter density on the other hand. And so then what we think happens is that neurotransmitter receptor density actually modulate the neural circuitry's response properties. So we can then build a little model of what the biological implementation looks like. So on the left, you see a model of the visual hierarchy, and you see that neurons take their input from a little region, and that is progressively pushed through the visual hierarchy that gets bigger and bigger. And we basically have two pools that we sort of simplify as an inhibitory pool and excitatory pool whose connections are actually modulated by these neurotransmitters. So what we have now is really different ways of looking at contextual mechanisms. So we have, and this comes down to the Mars 3 levels. And so you have the computational goal level. And the computational goal is really that you take vision into context. So vision is not determined solely by the stimuli, but it's also determined by the context. And to some extent, almost everything is determined by context. The algorithm by which we do this, I think, is divisive normalization. And we have done this in the visual space, but others have done this in memory and other domains. So this really seems to be a mechanism operating throughout the brain. And what we think happens is this is actually implemented by at least variations of different neurotransmitter densities. So to go back to my conclusions, what I'm trying to portray is that vision is a window into brain function. Context shapes vision. We think this is implemented in a divisive normalization algorithm. And these both biological processes rely on neurotransmitters. So I've been telling you all this, but I started with psilocybin and psychedelics. So how does this come back to producing hallucinations? Well, if we have these different, the goal, the algorithm, and implementation, psilocybin is a serotonin agonist. So it really acts on these receptors, which I think if we follow our logic through, should then change the divisive normalization computation and may potentially alter perception. And so this is really the hypothesis, right? So does psilocybin actually alter this algorithm, and does psilocybin then also alter perception? So this is another way of putting it. We have our simple circuitry model. If we administer, or if the participant takes psilocybin, we expect that to interact with especially the serotonin receptors. And from the literature, you can read which serotonin receptors would be mostly affected, but you can debate about this. But what we think here is that, okay, this very core circuitry that we have, they should function differently under the influence of psilocybin. So what we did is a randomized, double-blind, placebo-controlled study. We had several participants who participated. They went in 3 times. So on 3 different days, they came to the Spinoza, to the Netherlands Institute for Neuroscience, to participate in a neuroimaging experiment. And in all 3 cases, they got pills, always 2 pills. It could be 2 placebo, 1 times placebo, and 5 milligrams, or 2 times 5 milligrams. And then we put them in the fMRI scanner, and we showed them these stimuli that I've been showing you before. Now to really get back, so how did the subjects perceive it? And I'll show you a little bit of anecdotal experience. So one subject actually said, I hope this, he came in for his first session, so he didn't participate in the other 2 sessions yet. So we knew he had to go through 3. And on the first session, he got what he thought was a very strong doses, and he said, okay, I hope this is the 10 milligram. And because otherwise I'm going to get the 10 milligram later, and this 5 if this is 5, then the 10 I don't think I can manage. So this sort of gives you the subjective strength of the psilocybin drug that we gave them. It actually turned out, in retrospect, as experiments, we didn't know either. But in hindsight, it was the 10 milligrams, so he got lower doses later. But it was randomized, and neither the experimenter or the participant knew. Throughout the experiment, so psilocybin has a very long duration that takes into effect. So you take the drug in the morning, and then at 5 o'clock, the effect has completely worn out, so then they can go home. So this experiment took a day. But during the day, we asked them different questions, and this is a very subjective, about the subjective intensities. And these are the type of questions that we then ask, how panicked, how frightened they feel, how stressful they felt. And this is kind of a standard psilocybin or psychedelics questionnaire that people use. And you can actually sort of, at the end, sort them based upon whether this was placebo, 5 milligram, or 10 milligram. And in this graph, you see the subjective intensity as from the time of administration. So you see that if the subjective intensity actually increased for the 10 milligram doses, as relative also to the 5 milligram, and placebo has, not unexpectedly, a little placebo effect, but very little changes in the subjective experience. So this actually means that the drug actually does have an effect on this, how the subjects actually perceive the world. And as you notice, they do actually answer questions about their vision and about how they feel things distorted. So they do, have some distortions in their vision. And during this time, we also took neuroimaging experiments in this block and took behavioral experiments in the second block. So if we look at the raw functional imaging responses, or the raw fMRI responses, to 1 bar sweep, then this is what you see. You see the signal intensity as a function of time, and this is relative to when the bar passed through the population receptive field. And what you see is what I've shown you before, this characteristic Mexican hat function, and this is for placebo. Now, if you plot the same response when they're from the same brain, but just at the different times, for 5 and 10 milligrams, you see that the decreases in responses are actually altered. But the central response of the PRF has actually remained the same. So it's mainly those surround responses that are affected. And this is just looking at the raw signal. But we can also, look at the model. So the raw responses are affected, and how does the model interpret it? So we can plot the same thing. So now we fit our divisive normalization model. And if we do that, in particular look at the activation context, so the one that actually captures the surround effects, then you see that the activation context is actually decreased when you, as a function of the doses. So it's the strongest, as you saw in the slide before, with placebo, and it then decreases with 5 milligrams and 10 milligrams. And this is kind of what you would predict, right? So if the activation content is high, you get the suppressive surround. If it's low, you sort of start disappearing. So what we see is that the activation contents or the suppression is actually affected in a very dose-dependent manner. We can also, of course, look at all the other parameters. The model has many parameters. We can also look at the other parameters. I'm not going to go through all of them, but basically what you see is there's not much variation in the other parameters. So the other parameters are not affected. So it's really specific, the effect to this activation context is B parameter, which builds the surround. So what we see is that psilocybin affects the activation context. And so if we go back to our little model, then we think that psilocybin then particularly acts on the serotonin effect, and that actually fits with the model, right? So we actually saw that the serotonin H2-1A receptor correlated very much with this B receptor, and this is the one that is actually what we speculate is then mostly affected by the psilocybin. Although, of course, we cannot exclude that other neurotransmitters are affected as well. We also did some behavior, so they went into the MRI scanner and then took a little break and then did some perceptual tasks. And so now I'm going to show you the result of these perceptual tasks. And we actually looked at this Ebbinghaus illusion that I showed you before. And the basic question that they had to ask is which circle is bigger? So here's the results. And if you ask them which circle is bigger but there's no surround, then you get this gray response that goes to this typical psychromatic curve that you see with probability versus the size of the dots on the left and right side of the stimulus. And so what you see is this really stereotypical psychromatic curve. And I don't show the 3 doses here actually, but they're actually identical for the 3 doses. And now we can actually ask them the same question, but now with the surround, right? And then you see that even in placebo, there is a bias. And this is basically the illusion that I've showed you before. They underestimate or overestimate one of the circles. So this shift on the y-axis is basically the illusion. And this is on the placebo. And this is what should happen, what we know should happen. And then we can ask, okay, how does this change when we administer psilocybin? So this is 5 milligrams. And 10 milligrams. So you see that the illusion actually increases in strength. So psilocybin actually alters perception and is dose dependent, but it does it only when they're doing this Ebbinghaus illusion, so only when there's a surround present. And when there's no surround present, they have no problem doing this. So there's not dose affected, there's no effect of psilocybin. So psilocybin really affects context-dependent perception. And if you think that the drug sort of enhanced anxiety or made the system noisier, they can still do the task perfectly identical when there's no surround. So psilocybin also affects context-dependent perception. So really, how does psilocybin shed light on hallucinations? And I think this following hypothesis comes forth from this research, that psilocybin is known to affect key neurotransmitters, especially the serotonin neurotransmitters. It changes the neural computation, and specifically the computations that normalize how we perceive things in context. So in the algorithm level, it changes the activation context constant, which is really sort of there to perceive or to compute contextual computations. And what we also see is that psilocybin alters perception. So psilocybin disrupts how the brain uses context to interpret the sensory input. So what really then puts forward from this hypothesis about how we could think about hallucinations, that if the brain loses its contextual balance, hallucinations may occur. So very clearly on a neuronal level and on a perceptual level, psilocybin alters contextual computations, and if you cannot place things in context, we think or we hypothesize that that may actually give rise to hallucinations. This is all I wanted to say. I wanted to thank my group at the Netherlands Institute for Neuroscience and the Spinoza Center for Neuroimaging. Once again, I should acknowledge Marco Akiel, who is the PhD student who is leading the study, and also Thomas Knapen, who has been working on this closely with us. And thank you for your attention.
Thank you very much, Serge. If there's any question from the floor, I will take it first. Otherwise, yeah, there's one.
Thank you for a great talk. I had a question about the psilocybin. So if you give a group of people different or at the same dose, they might respond in different ways, same as people might respond differently to alcohol intake. And maybe some people have a proper hallucination and others don't. Did you see this? And can you tell from the fMRI responses how heavily they were affected, like as by the individual?
Yes, so that's, thank you for your question. I think we actually looked at whether the perceptual effect and the neuroimaging effect correlated between individual subjects. Now I showed you the group responses. And if you look at the correlation, there's a weak correlation. So that does actually look promising. We only had 12 subjects in the end, and we're actually collecting more subjects as we speak. So I think that is kind of minimal to sort of establish these correlations. But the very preliminary evidence seems to suggest that it correlates. So yes, people who may be affected more might also be affected more both in neuroimaging and in perception.
Thank you.
I also have one question, actually. I've been always thought this context in visual field has got to be a higher order of visual field. And your plot with fMRI and PET that showed different areas, and it looks like a V1 has the greatest correlation value there. Is that right? Am I interpreting the data okay?
So I think, so with fMRI, we cannot tell where it comes from. So I think it's actually some feedback of some areas. And I think the other thing is, if you look at the B parameter, you do see it coming back into other areas as well. So it's not like it's not there. In this Mexican head shape, it's most obvious in V1 because it's clear separation. As the PRF gets bigger in later visual areas, it becomes harder and harder to distinguish the two. The model still says, okay, there is this activation constant, but I think as in showing the raw time cursors, it becomes less obvious. You also see some compression, but it's also less intuitive to plot in this kind of fashion. So I actually think this happens later on too, but yeah, I didn't quite focus on that.
Thank you. Yeah, there's one other question.
Hi, thank you for your lecture. Can you suggest some strategic tips for global cooperation with your 1 or 2 experience?
Yeah, so I think what comes out of this is a very specific hypothesis, right? And we can only do so much with neuroimaging. So with fMRI, we look at bold responses or blood oxygenation level dependent responses. we cannot look at individual neurons, which individual neurons are actually modulated with this, so you can actually do this in animal models. I think if you look at post-mortem tissue, then you could actually say, okay, in these kind of clinical disorders, since we now have a target neuroreceptor to look for, is that actually altered? Is this neurotransmitter balance also altered in these psychiatric disorders? So I think this opens the door, really, or it gives an alternative route to explore, perhaps, or an hypothesis to say, okay, how can we see this further? And so we think this is under the influence of drug. Does this hold in the psychiatry disorders? Can we see signatures molecularly or in the neurotransmitter receptor densities in these disorders? And what happens at the neuronal level, because we are looking at this coarse population level, and the population is about 1 million neurons, but neural types differ. And so I think there's lots of scope to actually go a little bit more in depth. Now, this is not the only method that you can use to study hallucinations, so this is just sort of our little way. But I think if you put them all together, I think we can hope to gain more insight in what produces hallucinations, and also in what, what is the sort of the neuronal basis of these psychiatric disorders, and hopefully in the future also how we can actually help them.
Okay, unless there is more question, I think I would like to close the session, and thank you very much. Serge, please welcome. Please join. Thank you.
Thank you very much, and please give another big round of applause to our moderator and speaker. Now we will take another 10-minute break for the coffee and networking session. And please feel free to enjoy refreshments at the back of the hall. And also there are various programs in the lobby. And see you later. See you 10 minutes later. This session will be led by Professor Chang Yong-jang from Sookmyung Women's University. Please welcome him with a big round of applause.
Thank you for the kind introduction. I'm Chang Yong-jang, a leader at the Gene Therapy Research Center at Sookmyung University. It's a great pleasure to be here today. At our Gene Research Institute, we are collaborated with Stefan's group at the Ghent University, Belgium. And we try to develop a specific gene delivery system. For example, we want to develop external stimulus responsive gene therapeutics. We applied electric field to cancer research or cancer therapeutics development, and also Stefan Groups has specific fantastic synthesis technology of nanoparticle for gene delivery. Therefore, we try to develop electric field responsive gene delivery nanoparticle. And finally, we can specify this gene delivery nanoparticle at specific site, especially cancer or specific organ. That means that we can minimize the side effect of chemotherapeutics, and also we can maximize this drug effect. I'm going to introduce today's speaker, Professor Stefan. It's my great pleasure and honor to introduce Professor Stefan to this session, because Professor Stefan has made a remarkable contribution in the field of gene therapy, and we are truly privileged to have him with us today. Professor Stefan is the founder and director of the Ghent Research Group on Nanomedicines from Ghent University, Belgium. The research focus in his lab is on the delivery of biotherapeutics, especially nucleic acid and proteins for future therapeutics for lung and ocular disease and cancer. Together with the partners in academia and industry, his lab developed and evaluated delivery concepts and technologies of novel advanced therapeutics. Recently, his lab got the famous award from Nature, spin-off award. He has been a guest professor at various universities in Belgium, China, and Italy. Especially, he is leading the Journal of Controlled Release as an editor-in-chief. Okay, and let's move on to his talk. The title of his talk is Multidisciplinary Approaches for Precision Ocular Delivery of Nucleic Acid. Okay, Professor Stefan, please take the stage.
Yeah, okay. So thank you, Mr. Chairman. So good morning, or sorry, good afternoon for you, dear colleagues in Korea. First, my apologies that I cannot be with you today, but it's really my pleasure and also my honor to present you part of our work we are doing here in Belgium. Probably most of you know what nanomedicines are, and so it means that we design a kind of nano-sized, very small nanoparticles in which, for gene therapy, in which we try to encapsulate nucleic acids like genes, mRNA, and so. And of course, the aim is that when you encapsulate these nucleic acids in particles, that they can be easily taken up by the cells, where the nucleic acids can be released in the cell to, let's say, to express proteins or to knock down proteins for all kinds of diseases. So in our group, we have actually 4 teams working on different targets for this nucleic acids and for nanomedicines. So we have people working for nanomedicines or on nanomedicines for ocular diseases, for lung diseases. Also, we work a lot on nucleic acid nanomedicines for vaccination. And a typical example is the COVID vaccine, where you have the mRNA encapsulated in lipid nanoparticles. And we are also working in the field of this ex vivo cell therapies for cancer. Maybe important as well is that since rather recently, since 1 or 2 years, we also tried to install here in Belgium what we call a nanomedicine center, where we actually tried to advance, to make progress with advanced analytics to characterize in a very fundamental way, in the best way we can, the properties of these nucleic acid nanomedicines. Because of course you can imagine that the analytical characterization of such complex medication is highly important for clinical use. But today I will especially speak to you about our work in the field of the eye, so nucleic acids and nucleic acid delivery for ocular therapies. And actually the point is that already since actually more or less 20 years in our group, we are working on the following question. So we are working on the question, can we actually deliver nucleic acids in the retina, so this is the back of the eye here, after we inject nucleic acid nanomedicines in the middle, in the vitreous, in the glass, in the gel of the eye, in the vitreous of the eye. So can we develop nanomedicines which are intravitreally injected in the vitreous and which then move and transfect and deliver the nucleic acids in the back of the eye? And so this was our first paper on that. And I must say that I faced a lot of difficulties to do that, and I tried to just to summarize this to you. So here, one example, for instance, what you see is the following. If you inject nucleic acid nanomedicines, these nanoparticles in the vitreous, and in this example, we made use of a kind of lipid-based nanoparticle, where we in red labeled the EGFP encoding mRNA. So what you can see is that, and you go to the left here, so this is the vitreous, this is the retina, all these cells in the retina. So you don't see any green expression of the EGFP protein. And here you see a clear lining, so we inject in the vitreous a clear red single, which comes from the red labeled nucleic acid particles. So what it means that the nanoparticles, they do not have the capacity to go into the retina and to deliver the mRNA. In the opposite, in case you inject the same formulation, the same nanoparticles, here, that way, we call this subretinally, so below the retina, you clearly see expression of the green protein. The point is that these are very difficult injections and also dangerous because, of course, you can imagine that you are injecting close to the retina. So this route into a vitreal injection is much more preferred from this safety point of view. So we actually found a reason why these nanoparticles do not transfect the retina if you inject them intravitreally. And what we found is the following, and I was not aware of this, but ophthalmologists, they told us that between the vitreous, the gel of the eye, and the retina, you have this thin layer here. It's called the inner limiting membrane, which is a layer which is rich of collagen. So it contains a lot of collagen. And actually, we have shown that if you inject your particles in the vitreous, that they cannot transport, they cannot pass this collagen layer because that's needed to enter the retina and to transfect the cells in the retina. So we found that this ILM is a huge barrier for intravitreally transported particles towards the retina. And you can see this here as well. So here you see the retina. I lost my screen. It's gone. Can you still see my screen, please? Hello?
Yes, we can see your screen.
Yeah, so I don't see my slides anymore. Oh, maybe, can you still see my screen?
Yeah, we can see your screen.
Yeah, yeah. Maybe wait a moment, please. Yeah, I try to, yeah, I have some difficulties here. I hope you can follow, but you don't see my pointer anymore, I guess, isn't it? Can you still see my pointer? No.
Yeah, we lost your point.
Yeah. Okay, so I try to continue, hopefully. Yeah. So what you can see on this slide here is the following. And so you see the retina, and you see this red line, and then on the right side you see the vitreous. So you know all these nanoparticles labeled in the reds, they're actually stuck to the inner limiting membrane, so they do not enter they do not enter into the retina, which is a huge problem, of course. Yeah, it's, I cannot move it anymore. No. I'm sorry for that, but it seems my presentation hangs for one or another reason.
Okay, it's okay. Take your time.
Start again. No.
Stefan?
Yeah, hello. It seems I cannot move my slides.
Yeah, how about close your file and reopen again?
Yeah, that's what I try to do. Check. Oh, maybe it goes now. Do you see it now moving?
Yes, we can see.
Okay, okay, then we can continue. I'm sorry for this. And then you see my pointer. So you know from this slide, it should be clear to you that if you inject nanoparticles in the vitreous, they cannot enter into the retina due to this inner limiting membrane. And actually, so this also explains. So you know, at this moment we have one medication, which are viral particles, which are clinically used. They call it Luxturna, and they are in the clinic used for the treatment, the gene treatment of the certain disease in the retina. And as you can see, these particles are just injected subretinally. These viral particles are injected subretinally, because if you inject them in the vitreous, they cannot pass the ILM layer, so the ILM layer is blocking these viral particles as well. Okay, so besides this collagen layer, so besides this ILM layer, which is a huge barrier, we also observed actually that the vitreous itself, so the gel compounds itself, are a huge problem for nanomedicines. So what you can see here is a very simple experiment. So here we injected positively charged just plastic polystyrene beads in the vitreous. And you can clearly see that these particles, they do not move anymore. So each color is one particle. On the other hand, if you take negatively charged particles, you see that these particles are moving in the vitreous. So what does it mean? That if you inject nanoparticles in the vitreous, you should really optimize their surface properties to avoid binding of these nanoparticles to the vitreous itself. Because of course you understand, if the particles bind to the vitreous, they become completely blocked and do not enter the retina at all anymore. So these are all kind of challenges which we are facing here. And important to stress as well to you is the following, that, you know, I had many contacts with ophthalmologists, and they actually told me that, of course, the vitreous in the eye, it's really changing heavily upon aging. And just to show you a few, you know, some very excellent pictures actually. So this is here the vitreous on the left side, top. of a young person. So it's transparent. On the other hand, here below, you have the vitreous from aging people, from older persons. So you see clear aggregation of the compounds in the vitreous, which changes the structure of the vitreous. And also as another example, so this is here the ILM, so this collagen layer I told you about, the vitreous. In a child, in a fetus, it's very thin layer, while upon aging, this layer also becomes much more thicker. So what is my point? My point is that you can imagine that all these kind of aging phenomena you have in the vitreous, like this thicker ILM, the fact that the vitreous change in structure, that all these changing properties might have a big influence on what happens when you inject nanomedicines in the vitreous. And we should have this under control. So we have huge, let's say, challenges when we want to deliver modern medication like nucleic acids and proteins in the eye. And how to solve that, so our group is dealing with it. And it's important to stress to you that there are many ophthalmological diseases which remains important targets and which cannot be cured for the moment, or which cannot be cured. I give you a few examples. So Meesmann corneal dystrophy, so it leads to blindness, and this is a dystrophy of the endothelium. So here you have the epithelium of the cornea, the endothelium. Fuchs disease is another, an incurable disease at the moment. It leads to an intransparent eye, no vision anymore, due to diseases, dystrophies in the endothelium. The same, you have major challenges for eyes which are infected with acanthamoeba, parasites. Difficult to treat. Here we have another example of a corneal epithelium, a cancer growing in the epithelium layer, very difficult to treat. So just to tell you that at this moment we are lacking modern medication which can be used in the eye, like proteins and nucleic acids, and especially we are lacking suitable delivery vehicles, administration principles, delivery technologies to deliver those medication in the eye. So what I will try to do you today is to give you kind of short updates of, I would say, efforts we are doing here in our lab to come up with new technologies for delivering of nucleic acids in the eye. And therefore I have to explain you first the following. So in our work, in our lab, we have worked a lot on what we call photoporation of cells. So probably some of you know electroporation. So what you do with photoporation is you take your cells. So first we do this, or we do this in vitro, in cells growing in the 96 well plates. So what you do is you take your cells. Here are the nucleic acids in green. And what you do is you add a photosensitizer. So these are gold particles. So these orange particles are gold particles, they absorb light. And what you then do is the following. So you apply a pulse laser light. So very short pulses, nanosecond laser pulses. And these gold particles on the cell membrane, they absorb the laser light and they heat up. And as a result, you have the evaporation of water locally, which generate these kind of nanoscopic water vapor bubbles, so the purple or the vapor bubbles. And we have shown that when these vapor bubbles, when they actually implode, that you get small pores in the cell membrane which allow the nucleic acids to enter the cells, as illustrated here schematically. So this is photoporation, making pores in the cell membrane. This is electroporation. With no business, you make use of electric fields to put the cell membranes and ongoing to those things. So we have just for photoporation, but electroporation is not the topic of it. So this is the principle. So you have a photosensitizer like gold particle. It can also be a dope particle, or it can be another particle that absorbs light. You apply strong nanoparticles of the laser, and you get the evaporation of water. And these nanoparticles, as we call them, they switch for short term, nanoseconds, microseconds, and when they load, they get kind of radiation from the cell membrane. Okay. So here is the so on the left side you see just the pulse laser irradiation, and the right side you see the white dots here, which are gold particles. This black zone places were irradiated with laser also, and these are white scattering objects. These are those for a short time, which is improved as a light. Okay. So as I told you, so typically this was referring to that. So actually, we have made enough on this technology. It's called TRIS. We actually got the Nature Award last year for this technology. And I'm glad to tell you that even already we have colleagues who are actually ready to use technology for delivering all kinds of nucleic acids in cells into people. You know, I was wondering what can you do with these nanobubbles, these pulsations, not only in these sexualities, but especially in the body. And of course, you meant that I perfect place of laser points. So the question I got the following. So it's important for you that pulsed lasers as you using for operation technology, they're also used in the clinic, in the ophthalmology. So people are using them. So they're able to see pulsed lasers on one hand. Also in ophthalmology, they make use of photosensitizer that are for free, also ocular plasmids. So surgery people are making use in ophthalmology of all kinds of dyes to stain tissues, to make them visible during surgery. So there are also doing highlights. So we wondered if these technologies are used, these lasers, and also the ocular side, can maybe do nanobubbles in the eye? Can we do something with these nanobubbles for delivery nucleic acids and also surgery in the eye? And as I showed you some examples of today. So I showed you an example of the front of the eye, here it is, the cornea, the vitreous, and also the back of the eye. And our first question for following, can we deliver nucleic acids in cornea by nanosecond light flashes? And just to remove a little bit, the structure of cornea, this cornea is here, and it consists of a number of layers. So here you have the teloid, this is the epithelium, this is the oxygen of the cornea, 7 layers. Then you have the stroma, this layer here. And then very at the bottom, so this layer here, compact with the space. So this layer here is the single cell layer, which is epithelium. So we can deliver by photocopulation nucleic acid in the epithelium and also in the endothelium. And I showed you the results. So first we did epithelium. So we did the following with the slaughterhouse, and we actually cultured and so already both corneal epithelial cells, which applied by neurons, and we applied photocopulation. And then simple, a few one exactly 7 seconds. EGFP, mRNA, which is encoded in protein, and here we are what's called photosensitizer, which is polydopa slide. I will not go into the details, but you see clearly on the right side that these corneal cells start to access the green protein, clearly showing that formation allows mRNA in both of our cells. Then we took a next further. So we took an eye, whole eye, so slaughterhouse, 5 eyes. So we applied the sensitizer here on cornea. This is a ring to it, way. We positioned the eye that way in a kind of cup where we apply resolution of mRNA, and then we apply photosensitizers, now photosensitized lasers. After the treatment, we remove cornea and we analyze it microscopically. And what you can clearly see is that in the T cell, you can clearly see expression of the mRNA again. And then so what can be in vivo? And indeed, the animal can deliver nucleic acids in the epithelium by using the photosensitizer, by using lasers. So what we did is the following: we took animals, rabbits, we dropped the photosensitizer here. Sorry, just come again. Oh my God. Still see my slide, yeah?
Yeah, we can see.
Okay. So what we did, so we applied the polydopa on the surface of the eye of the rabbit. So we washed the free polydopa, and we applied the surface of the rabbit, the mRNA solution, and then the scanned, just scanned the whole eye. And what you can see is the following. So that's typical works, that's the scanning procedure. You can see it here, so the scanning of the epithelium with a pulse laser, right? So that's how it works, the rabbit eye. And here you can clearly see the following. So after the experiment, we actually removed, of course, again, the endothelium, and we analyzed it microscopically. I will not go into details, But that's what you see. So the epithelium here, this is the epithelium, this is the outside of the eye, the stroma and that layer. So in the epithelium, you can clearly see, of course, the blue colors, which are the nuclei of the cells in the epithelia, but also the green expression. And so green protein is expressed in the epithelial cells of this living rabbit. So clearly showing that photoporation allows to deliver nucleic acids in living animals. Okay. So then, of course, we do the next challenge, and we wondered, besides the epithelium, can we also deliver nucleic acids in the endothelium? Because there are many diseases for which it could be useful to have nucleic acid therapy in the endothelium. But of course, you understand that compared to the epithelium, this layer here is much less difficult to reach with photosensitizer. So here we just applied our photosensitizer on the epithelium on the eye surface. But if you apply these gold particles or polydopa particles, they will never spontaneously penetrate after local application on the front of the eye into the endothelium. It's too far. It's too, the cornea is a very stiff barrier. So therefore what we did is the following. So we injected our photosensitizer and our nucleic acids in this space. So this is the epithelium. Here this layer is the endothelium. This is called the intracameral space. And ophthalmologists, they often inject species, solutions in the intracameral space. So this is a kind of routine practice. So we injected our mRNA, our photosensitizer, and then we applied our pulse laser to parade here the endothelial cells. So that's how it looks like on the right side, ex vivo first. So we inject our material in this intracameral space. Yeah. And look, this is what you observe. So you clearly see expression of the mRNA, of the green protein in the endothelial cells of bovine eyes. And I must tell you that we could even confirm that in human donor eyes. So we got human donor eyes from our hospital. And if we inject intracamerally the photosensitizer and the mRNA, and we apply pulse laser light on the endothelium, you clearly see expression of the green protein in the endothelium of the human donor eyes. But also what we did, besides mRNA, we turned into siRNA, which knocks down the protein, and we took the siRNA for P16, which is a protein which plays a very strong role in the senescence of endothelial cells, which means if you can knock down it, you might induce the proliferation of these endothelial cells, which is of medical use, of course. And I will not go into details, but what you can clearly see is the following. So here you see the cells expressing the P16 protein. This is in a control experiment, but if you do the treatment, you clearly see much less expression of this P16 protein, so clearly indicating that we had a capacity to deliver siRNA as well into the endothelium of the cells. Okay. And then, of course, we took the challenge to go in vivo. Can we deliver nucleic acids in vivo in the endothelium of rabbits? And this was a very difficult experiment. So again, the same experiment. So we inject mRNA photosensitizer intracamerally of living rabbits, and we apply pulse laser light. And here you can see that. So here you have an OCT image of the epithelium of this rabbit. So here you have the epithelium, the stroma, the endothelium. So here you have the cornea. Here, these kind of small dots, these are gold particles, the gold particles which are injected in the intracameral space. You can see them. And look what we obtained. If we injected, first we injected fluorescent dextrins just to give it a try. And we clearly observed delivery of fluorescent dextrins in the endothelium of these living rabbits. And then we turned into mRNA. We inject mRNA in the eye of the rabbits, and we couldn't see any transfection of the endothelium. Actually, we found that the reason for that was that in the intracameral space, you also have enzymes, so nucleases, which fastly degrade the nucleic acids, which explain that we didn't see any transfection, any protein production in the endothelium. So therefore, what we did is we co-injected, besides the mRNA, an RNase inhibitor, and did the experiment again, and we clearly observed expression of this EGFP protein in that way in the endothelium of these living rabbits. So again showing that it is conceptually possible to deliver nucleic acids, mRNA as RNA, in the endothelium of living rabbits, living animals. So just for you to tell you, you can imagine that with photoporation you make use of a pulse laser. So this means you can easily select the position of that pulse laser. You can actually select which kind of cells you will actually irradiate, you will photoporate. And so in this context, we are currently working on the following project. So far we worked on the cornea, epithelium, endothelium. So now we try to transfect the liver cells which are present in the limbus of the eye. So the limbus is that region here, and in that region you have stem cells present which play a very important role in the growth of the endothelium and the epithelium. So what we try to do is, and by spatial selective photoporation, we try to irradiate this kind of circle here, so the limbus area, and try to selectively deliver nucleic acids in the stem cells which are present in this limbus. So we are currently working on that. We are currently doing this work, and I'm very glad to tell you that so during his stay in our lab, so Professor Junho Boon, who is now a professor in Seoul, he actually initiated this work, this initial work in our lab, this initial work on the photoporation of this limbal stem cells. So we are working together with our Korean colleagues in that area and try to make nice progress in this. So just to tell you, so this is the cornea, here we have the limbus, so we apply the light here you have the stem cells, so we try to deliver nucleic acids in these stem cells which are, in this limbus area. Okay, I show you another application of these vapor nanobubbles, which is first not really related to delivery of nucleic acids, but just to tell you very shortly. So you probably know eye floaters. So many people are suffering from eye floaters. So these are the kind of collagen aggregates which appear in the vitreous. So if you look into the sky, and now it's already blue sky here this morning in Belgium, so I also see some floaters in my eyes. So these are the kind of very dark regions which you see in your eyes. And many people are suffering from that, and especially Asian people. So the Asian people are suffering a lot from that. So what you have is the following. What is the reason? So in the vitreous of the eye, you have hyaluronic acid, you have water, of course, 99% of water, and a lot of collagen. And these collagen fibers, upon aging, they start to aggregate and form these actually aggregates. So here you can see that. So this is an OCT image of an eye, of a vitreous of a patient who suffers heavily from floaters. So this is the retina here, and here all these black dots are these collagen aggregates in the vitreous, which is very, very disturbing because it disturbs vision. So the current therapy is vitrectomy. It means so the doctor goes to the hospital and they actually remove completely the vitreous and they replace it with a saline solution. So to remove the floaters. Of course, this is invasive and also risky. And so therefore we considered another concept. So the idea we had was the following. Maybe if we inject a photosensitizer like gold particles and we make it to bind to the collagen floaters, one step. So here you have the photosensitizer, the gold bound to the white collagen floaters, and then we apply pulse laser light. We might locally produce nanobubbles on the floaters, on the collagen aggregates, which upon implosion might mechanically destroy and fragment the collagen aggregates, and in that way clear the eye from the aggregates. So look what happens here, and I hope it works. So on the left side you have, of course, in vitro experiments, you have such a human floater on which you have bound gold particles scattering the light. So I try to show the movie to you. So what this happens when you apply pulse lasers. You clearly see fragmentation of these kind of aggregates. And then so we also try to do this in vitro again. So we went to the rabbits. Of course, we have no animal model suffering from floaters. So first we injected collagen aggregates in the rabbit eye, and you can see them here. And so on this, I lost my pointer now. Where is my pointer? I cannot see my pointer anymore. But on the bottom, and so you clearly see the vitreous, and you see the aggregates present, and so in the vitreous. And so what you see, and then we applied laser light, and what you can clearly see on the left side, and you have these. I have it. Yeah, I have it here. Yeah, okay. Sorry, I found my pointer back. I hope you can see it. My pointer, can you see it? So here you have the floaters here, and after 3-7 pulses, you have destructed these floaters from the eye, from the rabbit eye. So we published this work in Nature Nanotechnology. I just tried to find my simple pen here. I tried to find my, in Nature Nanotechnology. You can find this work over there. Sorry. My presentation stops again here. I apologize. No. I apologize. My slides, the slides, my slides. Oh, I have it back here. Can you still see my slides, please?
Yeah, your slides doesn't move.
Does it move now?
Okay, move now.
Yeah, okay. So the point is the following. Of course, you can imagine that if you inject gold, that there might be kind of safety issues regarding the injection of gold. And therefore, what we did is, so, you know, ICG is a color, is a dye, an ocular dye, which is used in the clinic. And of course, you know very well that ICG is also injected intravenously. It binds to proteins because it's highly amphiphilic. And so therefore, we thought it will also probably bind, let's say, to collagen. And therefore, what we did in the kind of next step was, instead of injecting gold particles in the eye, we injected the ICG in the eye. And indeed, we found that it colors, it binds to the collagen aggregates, and that it can also actually implode and destroy the collagen aggregates from the eye upon applying a pulse laser light. This is also published in our Nature Nanotechnology paper. Okay, so let's go to another story. So in the middle of the eye, so you probably know this, this disease here. So this is, so in patients, and so in getting older, many patients actually, they might have posterior vitreous detachment. So what this means, in healthy vitreous, so in young patients, you have the vitreous which completely supports the retina, as you can see here. But upon aging, you have the vitreous which start to shrink. And so it means the retina, it detaches. So the vitreous, it detaches from the retina. But at certain places, you might have vitreous remain attached to the retina. And of course, if it keeps on pulling, it might break the retina, which is of course a huge issue. So at this moment, when this happens, of course, you have to go to the hospital immediately, and they actually remove it surgically. So they cut it, or they're also making use of these kind of enzymes, like this ocriplasmin and collagenase, to destroy here at that place the vitreous and to lose, and so to liberate the vitreous from the retina. It's very well known that these kind of enzymes are toxic for the retina, because of course these enzymes, if you inject them, they also penetrate into the retina. You can clearly see this on this slide. So here you have collagenase, which enters the retina. So you can clearly see that you have, compared to the untreated retina, you have a kind of, destruction of a kind of course, kind of deformation or this toxicity to the retina. So therefore, we wondered, what can we do in order, let's say, to cut, to cut these, to cut the vitreous here, to cut the vitreous here where it still attaches to the retina. And so the first approach we took was the following. So you remember that between the vitreous and the retina, you have this collagen layer. And I told you that this collagen layer is impermeable for nanoparticles because they are too big to move from the retina, from the vitreous to the ILM into the vitreous. So therefore we thought, look, in case we take nanoparticles and we immobilize collagenase on the particles, we might make a construct that is too large to penetrate through the ILM into the vitreous, protecting the vitreous. So it means we try to localize our collagenase here on that side, at this side, avoiding the entrance of the collagenase into the vitreous. I will not go into the details of that work, but if you have an interest, we recently published this in the GCO. And as a second approach, what we did was the following. So, yeah. Can you still hear me, yeah?
Yeah, it's okay. Go ahead.
Yeah. So please just tell me when I have to finish, because I lost the time a little bit. But, as a second approach, second potential approach for the potential treatment of this PVD, so posterior vitreal detachment, we reasoned the following. So what would happen in case you apply a photosensitizer in the vitreous and apply pulse laser light here in that region with the aim to make nanobubbles, which upon implosion might destroy the vitreous locally here in that area. Because you have the nanobubbles, so they upon the time of implosion, they might mechanically deforce the vitreous and degrade it. And actually that's what we tried. So what you have here is outside the vitreous. Of course, these are ex vivo experiments. This is here the vitreous, which is filled with polydopa particles, photosensitizer. And you will see what happens. You will see what happens when we apply pulse laser light. So when we apply pulse laser light, you create these bubbles in the vitreous, and you clearly, let's say, liquefy the vitreous. You clearly see that. And so you clearly see a kind of stream of nanoparticles here. So here they are immobilized so here they are immobilized, but here in that region they are moving. And so what we also did was the following. So I show this again. And so you have the nanobubble formation. It actually liquefies the vitreous, and you can clearly see that this liquefaction, because these particles are really moving here. And to confirm this liquefaction of vitreous, what we also did was the following. So we took our vitreous, we of course applied the photosensitizer, to make nanobubbles, but on top of that we applied a kind of probe, big fluorescent plastic spheres, 500 nanometer, which are in fact immobile in vitreous. They do not move, but what we hope that if we destroy the vitreous by these nanobubbles, that these green probe particles would start to move. And so we measure the diffusion coefficient of these green fluorescent polystyrene particles in the vitreous. So this is before the treatment, so the black line. We have a very slow diffusion, so no movement. And if you start to irradiate, to produce nanobubbles, you clearly see a movement of an increase of the diffusion coefficient, so clearly indicating that your vitreous becomes liquefied. Okay. So maybe to conclude my lecture, I will just give you some very final slides about the following. So one application which is more in the back of the eye. So I already told you before that it's highly difficult when you inject nanoparticles to get them transported into the retina because of this inner limiting membrane here, this ILM layer. So what we reasoned was the following. Assume that we apply ocular dyes in the ILM, like ICG, because there is a collagen there, and we try to locally apply the pulse laser in the ILM layer. So maybe what we can do is, so this is the ILM layer, this is the vitreous, this is the retina, this is the ICG molecules. So maybe if we apply locally our pulse laser, we can maybe locally apply nanobubbles here in the ILM layer, and maybe these nanobubbles may also porate this collagen layer, allowing the transport of nanoparticles from the vitreous into the retina. So I will tell you, if you have an interest in this work, you can go to these papers, but just to tell you that at this moment we have clear indications that by photoporation of the inner limiting membrane, you can improve the transport of nanoparticles from the vitreous, where they are injected, into the retina, which is of course also of high interest for the delivery of nanoparticles into the retina. So just to conclude my presentation here, of course there are many remaining questions at the moment. So I have not presented you solutions to you today for treating, let's say, ocular diseases and so on. I have just shown you our efforts in that direction, and there are many questions remaining, like for instance, so should we inject the photosensitizer in the vitreous, or should we apply them locally on the eye? So if you inject them in the vitreous, do they move from the injection spots to the target? These are all difficult questions, in fact, because in the end, the photosensitizer has to reach the target, either the collagen in the ILM or the collagen in the floaters or whatever, or the endothelial cells. So we are looking with the chemistry of dyes as well, and so to improve the affinity for ocular targets. So what about the residence time of these photosensitizers in the eye? So because of course ideally they should be clear from the eye. Of course, how to avoid the entrance in the retina to avoid toxicity. And in that regard, for instance, what we do is instead of injecting free photosensitizers in the eye, We try to couple them to polymers. We try to encapsulate them into nanoparticles. This to make them larger, to avoid that after injection in the eye they will go into the retina. And so we are currently following these kind of strategies. But it's highly challenging, I must say, because I will give you one example. If you encapsulate dyes into nanoparticles and your particles are not optimally, let's say, designed, you know, this can happen. So here you have vitreous, in which we have injected, let's say, nanoparticles which are not optimally designed. So you have massive aggregation of your particles in the vitreous, which is of course a major issue because it completely blocks, of course, the light, and you lose your vision. So just to tell you that what I have shown you today are just initial efforts just towards, let's say, new interventions and new treatments for ocular eye diseases. And maybe if there are younger people in the audience, I don't know, but this is the quote of our Belgian pharmacologist, very famous Belgian pharmacologist, Dr. Paul Janssen, who is the founder of Janssen Pharmaceutica, the major pharmacy company of Johnson & Johnson. So I would like to encourage you for that. So we all do this for the patients. So also at universities, we are working for them. So I would like to thank my colleagues, so especially Professor Reynald and also Professor Sauvage, and again also Professor Jungo Boon, because I'm so grateful that we have so nice contacts with Korea for the moment, and that we try to intensify our colleagues, our links with Korean universities, actually in multiple ways, I must say. And I'm very proud and also glad that our network in Korea is growing. And I'm also confident that for you in Korea, I have this impression that in most recent years, that also your links to European researchers is steadily growing and becomes quite impressive, I must say. So let's further build on the connections between Belgium and your country. And of course, this is our group of today. And of course, if you come to Ghent, we are living in a very, very nice old village, old city, I must say, a medieval city. And if you'd like to know more about our work, this is our website, so drugdelivery.be. My apologies for the technical issues here, but it's not always easy, of course, to present online. But I hope that you have understood the most important messages of this type of work we are doing. So thank you a lot for your attention.
Stefan, thank you for your insightful presentation. Because we have limited time, we cannot take questions from the floor.
No worries.
Instead, if you have some questions, please email me and I will forward to Stefan.
Yeah.
I have a short question. What time is it in Belgium now?
So I tried to know it. So at this morning, at this time, it's 8:08 in the morning.
Yeah, thank you. Thank you for joining us early in the morning, Stefan.
Yeah. So thank you again, Mr. Chairman. And so I'm looking forward to see all of you again. I must say that you are often in my mind, and I'm really looking forward to see you again and to further work together. Thank you for your invitation.
Okay. See you soon.
Thank you. Bye-bye.
Thank you very much, and please give a big round of applause to our speaker who have just joined us from long way in Belgium, and to our moderator. Now we will move on to our last and fourth theme, and it will be gene therapy. And this session will be led by Professor Jai Lee from UNIST, and please welcome him with a warm round of applause.
Hello, my name is Jai Lee from UNIST. First of all, I'd like to thank organizers for this wonderful global symposium. So I'm a PI for the one center of this global project. My center is focusing on development of the anti-cancer drug based on the synthetic lethality, especially the DNA repair genes. So in line with our project, today I host the Professor Myung-jae Kyung, who is a director of the Institute for Basic Science, Center for Genomic Instability. I briefly introduce Professor Myung-jae Kyung. He obtained PhD from the Brown University, and then he was section head in NIH for tens of years. And then he came back to South Korea He has been a director of the Center of the Genomic Instability of Institute of the Basic Science. He has been studying DNA damage repair and genomic instability for several decades. Recently, he developed a very novel and innovative anti-cancer treatment using the CRISPR-Cas9 system. So today he's talking about the new anti-cancer treatment. Let's welcome Professor Myung-Jae Kyung.
Okay, so thank you very much for invitation. So I'm the only one who between you and dinner, I guess. So I will do my best to finish as quickly as possible. So we are in the Basic Science Institute. Basically what we are doing is we try to understand molecular mechanism of DNA replication, repair, and recombination. That's what we typically do. But today I will not talk about any of those. Instead, I will just deliver one translational research that we started very recently and hopefully can lead some of the translation for cancer therapy. So I'd like to start with how cancer it is. So as you all know, cancer is the #1 death cause in Korea as well as in the world. And it is much higher than the heart disease or any other diseases. It has been about 3 different stages for cancer therapy that I will go through one at a time and how they work and then what's the problem to overcome in each stage of the cancer therapy. Let's just start with stage #1. So that's your first generation of cancer therapy. That's the radiation and chemotherapy. Many of drugs and radiations has been used to make DNA damages, and therefore rapidly dividing cancer cell no longer can survive. So that's the whole concept of this first generation of cancer therapy. So as you all know that many of these drugs that are still used in the clinic are listed in here. They come from bacteria, they come from plants, they come from many other sources. But the problem is, they can also kill normal cell. In our body, cancer is not just the only proliferating cell. There are other proliferating cells, and therefore, these type of treatment causes death in those proliferating cells. And therefore, there are so many side effects. So as you all know, that the cancer patients who treated the cancer therapy, they lose their hair, their immune deficiency. They also have a severe diarrhea that you all know. Let's think about the second generation. Second generation is a targeted therapy that we all like it. Personalized medicine, precision medicine. That's all coming from this second generation of cancer therapy. What it means is, let's find the cause of the cancer. Why cancer cell become a cancer? Find the cause and then target those causes. So I'm showing you one of the blockbuster drugs, the Gleevec, that targeting the chronic myeloid leukemia. So as you can see here, in 1960s, Philadelphia chromosome was found as a cause of chronic myeloid leukemia. And further studies found that Philadelphia chromosome is coming from translocation between 2 chromosomes, and that create a fusion protein, and that fusion protein eventually developed the cancer. So let's develop a drug that target those fusion protein. That Gleevec came out from there. It works beautifully. Typical chronic myeloid leukemia patient, their survival was less than 6 months. But after this Gleevec, they can survive more than 5 years and sometimes even longer. It's so beautiful, so that's the reason it was a cover of Time magazine too. However, it takes a really long time. Developing this with just one drug took 40 years to develop one drug. And also this drug only works for the chronic myeloid leukemia, also having this translocation. So therefore, not all the cancers can be cured. Let's move to the third generation. That's immune therapy. You probably heard the immune therapy a lot. It really works beautifully. So train your immune system to attack your cancer cell. It works. And however, the only problem is it does not work to all tumor. The desirable or expected cure rate is about 20% of the cancer. So less of 80% cannot be applicable by this immune therapy. So now, I'd like to remind you, cancer is a genetic mutation. So what that means is, during cancer development, in our normal cells' genomes, start to accumulate the mutation. During those mutation process, if the mutation happen to the tumor driver mutation, they become cancer. However, I'd like to remind you that in addition to those tumor driver mutations, there are many mutations are also accumulated. So this is a very recent, the Tumor Genome Project data. So as you can see here, in most of the tumors, they accumulate so many mutations. Okay, the number 0 in the table represents about 3,000 mutations. That means any patient comes in, take out tumors, you can find more than several thousands of mutations. And as you all know, the mutation means your DNA sequences are changed. Okay? And then you all know the CRISPR-Cas9. Probably in your laboratory, you use CRISPR-Cas9 every day. And I just want to remind you what CRISPR-Cas9 do. CRISPR-Cas9 can make DNA double-strand break in sequence-dependent manner. Okay? They recognize 21 nucleotides and cut the DNA. And this CRISPR-Cas9 has been used in cancer research. So mainly try to train your immune cell attacking your cancer. That's a major one. And other things, they try to change tumor driver mutation back to normal and to hope the cancer cell become okay. Now, I'd like to ask you a question. What is the most ideal cancer therapy? First, treatment should not affect your normal cell. Secondly, can it be applicable to all tumor types? So those are the two questions that may overcome all those cancer problems. So now I'd like to introduce first generation of cancer therapy again. I'm now showing you radiation therapy. Chemotherapy is the same. So as I mentioned, radiation therapy create DNA double-strand break, and therefore proliferative cells, they no longer can overcome those multiple DNA break, and therefore they die. However, side effect is that normal cells also be killed. And I have studied DNA repair in my entire career. In bacteria or yeast, when we give up 1 or 2 unrepairable DNA double-strand break, they die. Okay? We don't need like 100 break, just 1 or 2 unrepairable break. That's more than enough to kill the entire organism. And then I mentioned that when cancer cells become cancer, they accumulate multiple, thousands of mutations. Those mutations change in nucleotide sequence in your DNA. And also I mentioned CRISPR-Cas9 generate a DNA double-strand break in sequence-dependent manner. If you combine these two concepts together, we can design CRISPR-Cas9 that targeting mutated or changed DNA sequences in cancer cell and generate cancer-specific DNA double-strand break. And in such case, only cancer cell will die and normal cell will be fine. So like here. In radiation, it will break DNA, normal and cancer cell. But if we design CRISPR-Cas9, then we can only generate a DNA double-strand break in cancer cell. So we named this one as a Cinderella. So we decide to target insertion, deletion, mutation. The reason we chose insertion, deletion is to reduce off-target effect. That is a typical problem in CRISPR-Cas9 field. And so cancer-specific in the attacker. And we now call it potentially fourth generation of cancer therapy. So tumor has an in there mutation. Let's design CRISPR-Cas that can make a break in those in there mutation. Then we can eliminate only tumors by sequence-specific double-strand break. So first thing that we tested is whether enzymatic break, not by chemical, not by radiation, can kill the cell. So we have this laboratory human cell line that expressing restriction enzyme ASSI1. And this cell line, ASSI1, has a response element only upon tamoxifen, they moving into the nucleus. So we use this one to analyze the kinetics of DNA repair. But we just spontaneously express this ASSI1 in the nucleus by treating tamoxifen. As you can see in the red rectangle, entire cells are eliminated. So enzymatic DNA double-strand break, similar to radiation or chemicals, can kill rapidly dividing cancer cell. Then all those markers, DNA double-strand break markers are induced and cells are died, and they die by apoptotic cell death. Then how many breaks by our system? So it has been already published by others that this ASSI1 is a methylation-specific enzyme. So even though in the genome there's about thousands ASSI1 site, only about 100 breaks are expected to happen upon our system. So 100, that's more than enough to entirely wipe out the cells, cancer cell. Then we tested our Cinderella idea. So we chose 3 different cells. One is a normal cell, RPE cell, and U2S osteosarcoma, HCT116 colorectal cancer. And we analyzed the DNA sequences. We found more than 10,000 in the mutation that we can target by CRISPR in U2S and HCT116. And we designed 30 in the targeting guide RNA with a CRISPR. So as you can see in the middle panel, when we deliver 30 guide RNA that target U2S cells in their mutation, we can kill them all. However, in the same cell line, we deliver HCT116 in their target guide RNA, nothing happened. So there's no cross-reactivity because there's no sequence to cut it. So vice versa is the same in HCT116. So 30 is enough. Then we tested other cancer type, not just osteosarcoma, not just colon. We tested breast cancer, we tested blood cancer, liver cancer. So far, we tested more than 10 different cancer types. We could kill most of the cancer type with about 20 break. So up to this point, we used a viral delivery system. And however, recently we changed the system to the LNP particle. With LNP particle, now we can kill cancer cell with a 4 break. So here's an in vivo experiment by using LNP particle that capsulated Cas enzyme together with a guide RNA or messenger RNA of Cas together with a guide RNA. And then when we deliver them, as you can see here, HMix4 means HCT116 targeting guide RNA 4, targeting 4 guide RNA. MT50 is the one that targeting 50 location of the HCT116 genomic DNA. As you can see, tumors growth are retarded. Next, when we presented this data, so CRISPR-Cas9 field always ask, oh, what is going to happen if there's an off-target effect? yeah, it's a possible, but that's another additional break, so probably it will be fine. But to reduce that, we decide to use another CRISPR-Cas9 technology. That's a nickase. So CRISPR-Cas has a 2 enzymatic domain that can cut each strand of DNA. And by mutating one domain, you can generate a single strand break. So we decide to use a single strand break. By doing that, actually we can reduce off-target effect because a single strand break can be easily repairable. And one of the things for that purpose, we decide to combine another drug that's a PARP inhibitor, that's a blockbuster in clinic nowadays, that's coming from DNA repair field. So this slide here shows how the PARP inhibitor works. So when single-strand breaks happen, that happen all the time in your genome, the first responding enzyme is a PARP. And this PARP enzyme comes and tries to repair it. However, if you block PARP by inhibitor, then those single-strand break in cancer cell go to the DNA replication and create a double-strand break. And if you don't have a DNA double-strand break repair system, that's happen often in BRCA1, BRCA2 mutated tumors, and they can die. This is a concept of synthetic lethality, and PARP inhibitor works for that way. So we decide to use CRISPR nickase that just generates a single-strand break, and together with this clinically available PARP inhibitor. And indeed, when we try very little dose of olaparib, that's a PARP inhibitor, together with our nickase, we can kill those cancer cells, even though nickase by single-strand break does not cause any toxicity. That means off-target effect is very low by using this nickase. How about in vivo? In vivo system, same thing. In this cancer, olaparib PARP inhibitor doesn't kill cancer cell, and our nickase alone does not. But when we combine those two, we can suppress tumor growth. So with that, this is our platform technology called Syndela. So when patient comes to the clinic, what you need to do is simply do the two things: draw blood for normal cell, and then little biopsy of your tumors. And then we can just do the whole genome sequencing and compare them and how many targetable mutations we can find it. And with the algorithm we developed, we can actually ranking them what kinds of mutations could be targetable by Syndela approach. And with that, we design guide RNA and then pre-made Cas9, assemble it, and deliver to patient until tumors are gone. And if there's some resistance, then we always comes back to our original sequencing data, design another guide RNA to develop it. So that's our platform technology. So with that, I'd like to thank the people who has done. This one was mainly done in IBS and our school units by big collaboration. And now we are doing a collaboration with several hospitals in Korea and Asa Medical School and also Seoul National University. And then this technology is now tech transferred to the Cure Therapeutics, and they are now trying to see whether that can be really going for the clinic. Okay, thank you for your attention.
Okay, you want to take question from floor?
Thanks for a nice presentation. I have two questions about in vivo experiment. You have used nanoparticles by encapsulating with RNP and guide RNA. What is the loading efficiency into nanoparticles? Loading efficiency.
Oh, okay.
And second question is, in terms of in vivo experiment, your nanoparticles were intratumorally injected. Have you ever delivered them in vivo directly through the intravenous injection?
So for first question, I'm sorry that I do not know. So this was done by my collaborator, the Jinmyung Ju's lab. So they use this RNP particle that is actually designed by University of Texas Southwestern. So it's called a SORT nanoparticle. So if you, typically, so that I do not know, but intratumor injection, we did it, we did the IP, and recently we now start to targeting liver, so liver tumor, so we start to do the IV. All three cases it works. So I don't know how good this LNP particle, but it works beautifully. Yes.
Thank you.
I have a question too. So the cancer cells always find how to survive, right? So have you tried to target multiple like sgRNA at the same time?
Multiple
sgRNA like in their mutation?
Oh yeah. So current our method is trying to use at least 4 guide RNA. So for the safety, for clearly kill, I think we need to go up to 10 guide RNA to kill it. So that's the current our method. But the reason that we are applying this one with some combination therapy is to reduce the number. So we recently found that the radiation and this our Cindella or other chemotherapeutic agent or DNA double-strand break inhibitors, that's our all clinical trial at this moment, we can reduce number by half. So therefore, that's another strategy that we can combine it.
Wonderful.
I have one more.
Okay.
Regarding the previous question, when you do multiple knockout, especially more than 3 or 4 guide RNA, so you got the efficacy evenly, or depending on guide RNA, you have different knockout efficacy per each?
Yeah, so guide RNA choice is very important because so what we tested so far is if we design guide RNA very close each other in one chromosome, cell toxicity is low. Okay, so the choice of the guide RNA in different chromosome is always better, and then also large chromosome is slightly better. So that's so far what we know. And then also before we do the experiment, we always test that guide RNA for the correction efficiency by what CRISPR-Cas9 people usually do, and the correction efficiency should be more than 30% in vitro cell. And if it goes below that, the toxicity becomes really lower. So all those things need to be tested.
Is there any AI tool for designing multiple loci?
Yes. Actually, we are accumulating all this data by screening. So it's not me, but my collaborator, Sungwoo Jo's lab. He is now doing all this massive screen of guide RNA and then start to build up AI algorithm to pick it up the better guide RNA. So that's his specialty he's moving into.
Thank you.
Thank you. Any other questions? So I have a quick question.
Yeah.
So as long as the guide RNA and Cas9 survive inside the cell wall in vivo, then that persistently generate double-stranded break kill the cancer cell. Then how long the Cas9 system lasts once you inject the system?
Yeah, so protein typically lasts about 2 days. So that's so far what we can detect by ELISA or Western blot. And then when we add the mRNA, so now we are testing mRNA survivor in the mouse and especially in liver. It goes around 3 days. So if mRNA stays about 3 days, that means protein will stay about 5 days. So that's approximate that we can estimate at this time.
Okay, thank you.
Okay.
And if we don't have any more questions, then it's finished. And let's thank Professor Myung again.
Okay.
Thanks, Professor and moderator, for the excellent presentations and discussions. Please give another big round of applause to our speaker and moderator. Now we have concluded all sessions from the European region, And we'll move on to the closing remarks to officially end today's session. I'd like to invite one of our distinguished guests to the stage for the closing remarks. Let me invite Dr. Kyung-Gyu An from New Drug Development Center of K-MediHub to the stage for the closing remarks. Please welcome him with a big round of applause.
네, thank you all. Thank you all for attending the Advanced Bio Global Joint Research Center Symposium 2025 and staying with us until the final sessions. With the full support of all PIs and participating researchers as well as all staffs for this JRC program, we were able to organize the excellent symposium featuring outstanding speakers and to conclude it with successful networking among JRC members. Next time, we'll carefully review the overall performance of this symposium and come back with even better programs. Once again, we would like to express our sincere gratitude to everyone for your dedications and participation. Thank you very much.
Thank you, Dr. An, for your warm remarks. This officially ends today's session, and we would like to extend our sincere gratitude to all of you who have joined us until today. Advanced biotechnology is now advancing in an unprecedented speed. We hope that this gathering has served as a meaningful start point to lay the foundation for new collaboration research and to strengthen our sustainable cooperation. With that, we officially conclude all programs of the Advanced Bio Global Joint Research Center Hub Symposium 2025. Thank you very much. 네, 감사합니다. 이렇게 오늘 공식 일정이 모두 끝이 나고.
That concludes the official schedule of today's event, and now the lucky draw event. The much awaited lucky draw event, and we are going to give prizes to 2 people today. And once again, you cannot use the name tag again, and I'm repeatedly mentioning this because there are always people that forget about these things and lose these opportunities to win wonderful prizes. So if you haven't issued a new name tag, please make sure that you get a new name tag, and Dr. An Kyung-gyu will give the new gifts and prizes to the winners after the lucky draw event. And please understand that yesterday's winners cannot receive the gifts once again during today's lucky draw event. And I believe that we're going to do or select the winners based on the same method as yesterday. Let's see who will be the winner of today's event. I think I see higher numbers compared to yesterday. Do we have 140? Do we have number 140? Congratulations. We have the winner of a smartwatch. And that was the first winner. And again, do we have 153? Do we have 153? Yes, we have 2 winners. This has been the fastest lucky draw event I've ever watched, and the gifts will be provided by Director An Kyung-gyu. Please give the winners a big round of applause. Thank you for staying with us until the end of today's event. And please look at the camera in the center. Let's take a celebratory photo together with Dr. An Kyung-gyu. Please give the winners another round of applause. Thank you for being with us for the last 2 days. Have a safe trip home. Thank you very much.